Okay, so I'm just going to go over some of our current and future actions that we've been working on as a steering committee and as a working group. I also want to mention that although the working group was established in 2008, now more than ever, we're really in a great position to be engaging and connecting with our partners. In the past year, we have established a leadership comprised of a steering committee, a chair, and a coordinator. We published a website, and I encourage you to go check it out if you haven't yet. We've created a contact email account for the working group, which goes directly to me. We've reactivated the working group's listserv, which is how we send out announcements about meetings and provide other information. It's also there for the members of the listserv to use, and you can use it to send out information as well that's Cuckoo related. We've created a working group Google Drive. The access is currently limited to the steering committee, but we hope to eventually have some documents in there that will be available to the public. We've also established a regular meeting schedule for the working group. We have it twice a year. We have it in fall and in spring. We also have created a schedule for the steering committee meetings, which is 4 times a year, 1 every season. And in the recent, some of our recent meetings was the one last November and then this one last spring. We record and upload our working group meetings to our website, and Peak, which is our co-host, they upload it to their YouTube channel. And as you saw, I was setting up the recording for this meeting as well. We also have begun drafting and developing working group sponsored outreach and resource materials. For example, we identified priority research questions and topics, and we put that into an infographic. And we also are working on a handout on range-wide breeding habitat restoration recommendations that's being developed by Jay Carlisle and others. And we've also conducted an information analysis to identify priority actions within our 6 subcommittees. And then we put those actions into a strategic action plan table. And here you can see our action plan table. And we actually put this up on our website. And we just encourage people to go there and look at it. And if you would like to volunteer to oversee some of the subcommittees or some of the actions, or if you want to suggest other goals or objectives or actions that we should include, then you can either email me that information, or you could actually input that information onto our shared online action item table. And we haven't done something like this before. I don't know how messy it will look to have a shared table like this that's open to the public, but we're just going to hope for the best and see how it goes. I just wanted to note that if you do that, please make sure that your edits are marked as suggestions. Okay, so we have a lot of great things that are happening with the working group, and I just wanted to let you know where you can find that information. Like I said, go check out our website, attend our working group meetings like today. You can sit in on our steering committee meetings. You can email me and ask me when they are, and I can send you the Zoom link. You can also request steering committee meeting minutes. And you can read working group meeting programs. I created one for our meeting last spring, and I will create one for this meeting, and I hope to do it for every subsequent meeting. And it summarizes all the content that was covered in the meetings. So I'm going to now talk about some of the things that we are involved in. One of the things that we are trying to do right now is to secure coordinator funding. We feel that really a coordinator position is vital to the success of the working group. The coordinator provides a consistent point of contact and is really integral to sustaining and expanding the working group's efforts. I am the first working group coordinator since the working group was established in 2008, and I've been volunteering in this position since last December. Jay Carlisle, Jenna, and I have prepared a coordinator position justification, which has been reviewed by the steering committee, and we have started to disseminate out to partners. We were hoping that maybe we could get some end of year funds from state and federal wildlife agencies, but we were unable to locate that. We are exploring other funding sources like BLM's T&E recovery program, but we just really want to mention to everyone that We would love to have some suggestions on funding sources. You can put it in the chat or you can email me. Okay, another thing we're working on is creating a logo. I think it's important that the working group has a logo to make all of our documents, emails, etc., look more professional, and just for us to be on par with joint ventures and other working groups. This logo is still a work in progress. I'm still tweaking some things here and there, but I hope to revise this logo and have several versions available to the steering committee to vote on at our winter steering committee meeting. And if everything goes well there, then we'll have the final logo design rollout at the spring meeting. Okay, another thing we're working on to sort of formalize the working group is drafting a charter. Jenna and I have developed a draft charter, like I said, to formalize the working group's goals, objectives, activities, composition, rules, operating principles, and rules. For example, right now, we really don't have any method for nominating or selecting steering committee members. The steering committee has reviewed this charter, and we are currently revising the charter based on their feedback. And we plan to review this charter in detail and vote on its adoption at the winter steering committee meeting. Okay, another thing that we're working on, and I'm really, really excited about this because this is a long time coming, is the development of a centralized survey database. Partners at the U.S. Fish and Wildlife Service are developing a Western BPS-wide centralized database for Cuckoo survey data. And they projected that it will be fully operational and live in about a year and a half. Kristen Dillon, who's really been taking the lead on this, along with Megan Conway, is not able to attend the meeting this morning. And so she sent me this emailed update, and I'm just going to read it to you all if you don't mind. So this is what she says. The database development team recently sent us a trial version of the ArcGIS online database that they have developed to accommodate the data structure. They have asked us to test it out in ArcGIS online and field maps, examine the structure and relationships they have set up, and provide feedback. We currently are going back and forth with them to tweak things about the relationships and data entry forms. Once we iron that out a bit more, they're going to try to merge historical data into the database. They have asked us for polygons of field sites. In general, shapefiles are not submitted when annual survey data are provided to the service, but many projects consist of surveys repeated annually at the same study sites. And then she says, if anyone on the call or on the meeting has shapefiles or KMZs with their study site polygons that they're would be willing to send me, that would be extremely helpful and very much appreciated as we continue to try to develop a centralized database. And then she gives her email here, which is Kristen_Dillon@fws.gov. And I just wanted to mention that the contact information for all of our steering committee members are on our website. And so if you don't have time to write this down right now, then you should just pop over to our website and get that information. Okay, another thing that we're working on is building a Cuckoo literature database. Megan Conway with the Fish and Wildlife Service is migrating documents from her listing and critical habitat folders into Zotero. She has generated a bibliography for us that can be reviewed by the working group for accuracy and completeness. And we're going to be sending this out to the working group pretty soon for review. And we're also compiling Cuckoo literature into a shareable folder, actual papers and reports. And right now, that's only accessible to the steering committee. Okay, so we're also working with the Road to Recovery initiative. The yellow-billed cuckoo, both the eastern and western populations, is one of the 4 Road to Recovery pilot species. And we have been engaging in Road to Recovery by hosting, for example, an informative 2-day virtual symposium in November 2021 in coordination with their launch, attending the Road to Recovery Species Working Group engagement sessions, and coordinating with their social scientist, Holland, who will be presenting a little bit later, and also other staff. Also, Callie Stanley, who is on our steering committee, was awarded 2 R2R grants to begin work in the South American Gran Chaco to study yellow-billed cuckoos and neotropical migrants more broadly. And she hopes that plans that her fieldwork will begin this winter. It will contain 2 major components. The first one is surveys to establish the distribution of neotropical landbirds across the Gran Chaco of Argentina and Paraguay, And 2, a Cuckoo focused tracking study to look at the influence of forest cover on Cuckoo movement and overwinter survival. So Callie is now going to provide additional information about our work with R2R.
So yeah, I mean, Hira kind of explained our relationship with R2R. We're like kind of a pilot species, an unofficial, official pilot species. But yeah, we're really excited to be working with Road to Recovery. One of the things that Road to Recovery has started working on or developing, you know, we just had the working group workshop, I guess a couple months ago. The next step is in the Road to Recovery, for the Road to Recovery working group, is to put together an in-person meeting to try and bring together members of definitely the pilot species working groups, but also other working groups to be able to do an in-person meeting. And so right now we're still at the early stages of planning the meeting. No dates have been finalized. There is the potential for having, I mean, March and August has been thrown out there, but that's, you know, very preliminary. But we are at the process of putting together potential topics to cover in this in-person meeting. And so right now they have some general themes that they're thinking about for the meeting, but other than that, we haven't finalized anything. And so what I wanted to do today was just see if there was any specific topics that members of this group thought would be helpful to have at this meeting. And this meeting is really designed to build off of the workshop we had earlier this year and provide some more concrete opportunities for working groups to advance. And so any topics that we think could help our working group put together, you know, a conservation plan for cuckoos would be, you know, fair game to include. And so some of the major themes that have been discussed are building connections. So like within our group, like our working group or outside, increasing capacity, just the R2R process in general, if people felt like they wanted more information about what that meant. Organization, some of like our group is one of the groups that has quite a lot of organization, and a lot of the stuff that has been presented by Hira earlier today is included in that, like our databases, action plan, but how else can we organize stuff, or the group, the structures of the working group, and then data analysis. So more, if people had specific methods that they wanted to discuss or have discussed or thought about. So yeah, I just wanted to open it up very briefly to see if anyone had any thoughts.
So folks can unmute themselves and just chime in here if they have any thoughts about what would be some specific topics of interest for discussion.
Paul Schmidt just put in the chat that the Road to Recovery will be sending out a survey to species working groups and others to get input on topics, outcomes, etc. for the workshop conference that Callie is talking about. Thanks, Paul.
Yeah, so I just thought it would be helpful now amongst us to see if there were any topics that we agreed on or collectively thought would be important. But if not, we've also included in the Padlet that we're going to be using for the afternoon sessions, there is a column at the end that has Road to Recovery, a place where we could discuss it, aside from like the surveys that are going to go out. And yeah, I guess that's all I was mentioning.
Yeah, and I can put that link in the chat and then you just follow that link. And as Callie said, it's the last column on the right-hand side and you just hit the plus button and you can add your thoughts and ideas. And we'd love to hear from everyone.
And there's really no wrong answers. Like we'd like to know what people think is maybe missing, or if there's any topics we'd like to learn more about. Look forward to hearing from people. In this time, everyone is welcome to go to the Padlet and start adding suggestions about the R2R meeting or whatever.
Yeah, I placed it in the chat along with very long, detailed instructions about how to use the closed captioning, but the Padlet link is also in there. Also, if you want to take a quick little bathroom break. I'm going to call Hira and see what's up. She does have her next presentation in the shared folder, so I could open it up, but I know she wanted to give a brief little introduction to the morning sessions.
Hi, I don't know, sorry. We're using our co-host Peak account, and it keeps on saying Zoom is kicking you out, and I hope that this doesn't keep on happening too much here for the morning session. Luckily, Kristen O'Hara, who will be moderating the afternoon session, and she'll be dealing with that directly from her account. Okay, so I'm now going to move on to talking about our automated recording unit researcher meet and greet, and I'm going to upload my presentation for that. Okay, so like I said, I'm going to just quickly give a summary of a meeting we had this last September. So first of all, I just wanted to mention that in our action plan table, we have a goal that is identify effective and accurate methods for tracking, surveying, and monitoring cuckoos. And one of the objectives is to investigate ARU use, automated recording unit use. And one of the action items under that is to promote ARU use and accurate data processing. And so Nick Beauregard with the Southern Sierra Research Station and we decided to co-host a virtual researcher meet and greet in September. And first we sent out an invitation on our listserv, on the working group listserv, to join an ARU researcher email group. And we got about 25 respondents. And then we sent out to that group an invitation to join the meet and greet. And then we had the meet and greet. The goals of the meet and greet were to foster collaboration among researchers using ARUs to study cuckoos and their insect prey, understand the various methods used for ARU deployment and recording processing, identify advantages and disadvantages of existing protocols, encourage consistency in ARU deployment and acoustic recording processing where possible, encourage coordinated cuckoo acoustic monitoring and data sharing, and help to advance science on ARU use and applications. And we had 2 guest speakers, and we had 10 project-based presentations, and the format of those were, sort of a lightning talk format, just like we're going to have this afternoon. Most of the folks were talking about projects that were located in California, but there were also projects in Southeast Arizona, New Mexico, some proposed work in Southern Colorado Plateau, and in Paraguay, which is the work that Callie is involved in. The ARUs were deployed in a number of different habitats, including like restored sites, remnant riparian areas, native riparian woodlands, zero riparian areas, riparian agricultural matrices, and then Callie will be doing work in like tropical dry forest and other habitats in Paraguay. Some of the questions that folks were looking at answering were, for example, just, are there birds out there? What's the occupancy of these areas by cuckoos? Also, what's the breeding status of the cuckoos, the population sizes in a particular area, the population distribution across the landscape, how cuckoos were using various habitats, the efficacy of restoration efforts. For example, when you restore a site, do you then attract cuckoos to that area? Also, people were looking at the efficacy of different ARU use. They were comparing how different ARUs actually performed in the field. Folks were doing a number of different concurrent research projects. For example, folks were also doing just regular cuckoo protocol surveys. They were doing nest searching, and they were also completing surveys for other species of conservation concern. After all the presentations were completed, we had a discussion. It was a very lively and interesting discussion. It actually went 30 minutes over what was scheduled. And we just very, very loosely kind of tried to address these 5 discussion questions, which I'm going to sort of go over real quickly. One is, what are our shared goals? What information, tools, or products are most urgently needed to achieve those goals? 3, how can information be efficiently shared? 4, Should we develop a product to provide guidance on ARU hardware, deployment, processing, et cetera? And 5, how do we identify and engage the broader ARU community outside of the folks that were attending that meeting or that have responded to that they want to be on the email list? You can go to our website to find more information on that meet and greet. On our website, we have the meeting goals, the agenda, the chat notes, a PDF of the discussion Padlet contents, PDFs of selected talks. Not all talks were marked as available to the public, so we just have the talks on there that could be shared with the public. And also a link to the video of the discussion. Coming soon, I plan to put a summary up there, something that's similar to what is in this presentation, and that's only after I have it be reviewed by our participants. And then hopefully next spring, we'll draft some guidelines for ARU use in Cuckoo research. We'll be working with Nick Beauregard on this. And also we hope to have some acoustic monitoring presentations at our spring meeting. Okay, so in the next few slides, I'm going to sort of summarize what was discussed in the meeting. And I'm going to try to go through this slowly. It's a lot of information, but I think it's really useful information. And hopefully I don't lose connectivity. So one of the things that was discussed is acoustic recording units. And there are a lot of different types of acoustic recording units out there. And so one of the things we're talking about when we were discussing the different ARUs that are out there is that, maybe a good starting device for folks is AudioMoth. But some folks thought, well, the wildlife acoustic units are maybe more customizable. People also mentioned that they felt that ARUs are pretty durable. AudioMoths come with cases that seem to work well. Someone even mentioned that the SM4 unit survived fire and floods in one study area, which is pretty amazing. These different units do have different detection rates. And the Western Foundation for Vertebrate Zoology did a study comparing AudioMoth to Zoom H2N, and they found at their study site, at least, that the Zoom H2N actually outperformed the AudioMoth. However, many of these units are probably pretty effective at capturing vocalizations of birds within 100 to 200 meters. Some of the questions that were raised in our discussion includes the question of what is the detection range of each device? We'd like to have more information, more specific information for each of these devices. Because we really want to know how to maximize our coverage while minimizing the number of units deployed. We want to try to buy or use as few units as possible, but still be able to cover a large area. Okay, we also talked about recording times, and really our discussion was breeding ground focused. Most of the studies looked at the period of June to August, and they would run their devices daily from anywhere from 3 days to an entire season. And you can run the devices continuously during that whole entire period, or you can run them at set intervals, like at 10-minute intervals. You can have it turn on for like a minute. You can also have your devices recording across all 24 hours or at set times. And some folks just had the device turn on and start recording during the time period 2 to 4 hours after sunrise. Some of the things that we discussed is the fact that the occurrence and types of vocalizations of cuckoos vary within the day and across the season. So you have to think about that when you're setting up your recording times. Okay, another thing that we discussed is the spatial distribution of units. How far apart should they be? What's the configuration of the devices out on the landscape? I think in general, people were doing it every 400 to 500 meters, but it was sort of difficult for me to glean that from the presentations. I think they were trying to do that because that's sort of similar to what's in the survey protocol in terms of where you stop every 500 meters or so to play a cuckoo vocalization when you're surveying. So they're kind of mimicking that with the ARUs. So some of the discussion points is that it's really valuable to do a pilot study to look at the detectability range of your devices at your specific field site before you actually start the official study. And this is really because the spatial distribution of your devices is going to depend on so many factors. And I'm just going to quickly go over some of the factors that were mentioned. Like, what are your study questions? The amount of devices you put out there and how many you put out there is going to depend on, for example, are you just looking to see if there's even one cuckoo out there? Are you going to try to actually figure out how many cuckoos are out there? It also depends on like the unit type, the habitat type, and the spatial configuration of that habitat, the ambient noise that you're going to have to deal with, your recording frequency, the target species, which in this case was, cuckoos, and the life history of the target species, the size of their territories, their mobility. Are you looking at birds that are just migrating through an area, or are you looking at areas or birds that are territorial within an area? That's all going to affect how you set up your study design. And also, the vocalization occurrence. Cuckoos don't vocalize the same at the same rate at different times of the day. Maybe they vocalize more in the morning and not so much in the middle of the day. And also you have to think about the abundance and rarity of your species. If there's hardly any there, you might have to put more units out, for example, to try to actually capture some birds. There was some discussion about the fact that you do encounter some issues later on, that you didn't have the most appropriate sampling design to start out with. For example, maybe you had your units too close and you got some double sampling. You can actually correct for some of this during the post data collection modeling. Okay, I'm gonna now talk about frequency range. It looked like when I was looking at the presentation that most folks, the sampling rate for most folks was somewhere around 16,000 to 24,000, with I think most folks focusing in around 24,000. Some of the discussion points included the fact that ARUs are really effective at capturing multiple call types of the cuckoo, like the coo, cuck, help, and alarm calls. Coo calls in particular are pretty consistent. However, they're in a low frequency range that overlaps with doves and owls and other species. Alarm calls are also pretty consistent in their structure. However, contact and count calls are variable. They vary in structure, in the number of notes, the space between notes, etc. It's kind of cool to hear that ARUs have recorded calls of young in the nest and fledglings. Also, if you set the correct sampling rate, it looks like, acoustic monitoring for insects for the cuckoo prey can be done concurrent with acoustic monitoring of cuckoos, which I think will really lead to some interesting studies out there and findings about diet. Some questions that were raised include, how do cuckoo vocalizations differ among individuals and across the cuckoo's full annual cycle range? Well, let me go back. I wanted to say one other thing about that. For example, we think that they don't vocalize as much on the wintering grounds as they do on the breeding grounds. And, how do the actual types of calls differ between breeding, migration, and wintering? And that's going to affect what you're capturing with your units. Okay, let me go over some of our discussion about acoustic data processing. So there's a lot of different software that folks were using to process the data. Some were user developed. Folks use something called Kaleidoscope, BirdNet, Arbamon, etc. Things that we were talking about was that a lot of these software programs successfully modeled cuckoo calls. But in general, I think folks had to aggregate other calls because they're really difficult to differentiate. One of the questions that came up in our discussion was, can you determine breeding status or occupancy during the breeding season from just using these ARUs? And the answer that came out was yes. And that's by analyzing the occurrence of these different call types, like the occurrence of the coo calls, the calp calls, etc. Also, we can compare the acoustic monitoring results to results of concurrent nest searching and protocol surveys, and that kind of helps give you some of that breeding information. And you can also sort of follow the guidance that's in the survey protocol, apply that to ARU studies, which is basically that if you get 2 or more detections on the same ARU unit within 10 days within the breeding season, then you can call that individual a breeding individual that's occupying the site. Folks mentioned that occupancy models and species detectability models are useful in addressing data complexity and controlling for variation in the soundscape across the landscape and through time. Now, the rigor of your results is really going to depend on your study design. For example, whether the timing of the ARU deployment coincided with targeted activity. For example, if you're targeting breeding birds. Also, whether the timing of ARU deployment coincided with concurrent research, like protocol surveys, that adds some more information to kind of do a QC of your ARU data. And also how well your recorders performed and the accuracy of your analyses. Okay, we also talked about budget. And folks were mentioning that you need to not just factor in the cost of the units when you're writing up your budget, but also the field labor costs and the data processing and analyses. This is because the data processing can be very, very time intensive with an initial steep learning curve. And then of course you're going to have some, subsequent improved efficiency with increasing experience and skills. But folks thought, it's really beneficial to collaborate with folks experienced in processing acoustic data to try to reduce this time that it takes to learn how to use the software, and then reduce your budget as well. Conservation Metrics Incorporated, which had several folks present during our meet and greet. They mentioned that they're a for-profit business that can process and analyze acoustic data for clients, and they've been doing this, I guess, for a decade or so. Someone also mentioned during the meeting that an audio moth with its case, with its protective case, costs about $150 per unit, which I guess is cheaper than some of the wildlife acoustic units. And also, I think this is really important to mention right now, because if you want to use ARUs, you need to order them 6 to 9 months in advance. That means if you're thinking, oh, I want to use ARUs in my study next summer, you're going to have to put in your orders right now. I think you can probably get around some of this if you build your own units, and I think some folks are building their own units. Okay, so we talked about the successes of using ARUs, and ARUs, they are successful in detecting cuckoos and identifying breeding birds and identifying occupied habitats. So that's really great news. But folks experienced a lot of challenges too with this technology. People experienced equipment failures, classifier performance issues, software learning curves, and false negatives from similar sounding species like mourning dove. The fact that a number of calls are difficult to differentiate from each other. Also the fact that different ARUs have different detection rates. But they had to contend with lots and lots of ambient noise. And that there was a limited amount of true positive calls available to train their models, meaning that there were few calls of just cuckoo sounds that didn't have all of the sort of background noise that has been identified as truly being a cuckoo call, that there was a limited availability of those to train their models. So we identified 3 needs, and I'm just sorry, my throat is a little sore. I'm gonna take a sip of water. Okay, so one of the needs is to create a centralized database. Folks would like to have a database of manually annotated sound files of cuckoo vocalizations and background noises from the full annual cycle and from a diversity of geographic areas, habitats, and hours of the day. And also they thought it would be great to have sound files of true ambient noise without any cuckoo vocalizations, as well as AI algorithms. And this would help folks build an effective and efficient call detection and classification model. And we talked about the possibility of us hosting this database, maybe even on our website. Also, folks thought it would be useful to have some standardized deployment and data analysis guidelines for studying cuckoos. They thought this would be useful, even if these guidelines were really general and not very specific, because there's so many things that you have to contend with and think about on a site-specific basis that we probably couldn't have very specific guidelines. But maybe we could have more general guidelines that could still be useful for folks. Until this is drafted, it was recommended that folks visit the KITS's lab website, and we can put this link in the chat later. Also, we thought it would be useful to identify critical uncertainties and also then design studies to address those critical uncertainties. So some of the next steps include, us trying to address those needs I just mentioned by communicating and collaborating with other researchers involved in acoustic monitoring. Maybe, we'll continue compiling a list of researchers, and maybe we can see who's using ARUs already in sites that we think Cuckoos might be, and we can maybe use some of those existing acoustic data sets. We also want to help promote data sharing. Research to researcher, maybe on our website and on other websites. And during the researcher meet and greet, a number of folks mentioned that they're happy to share their data with those who are interested. We also would like to share other information and results of studies, for example, in scientific publications, and also hopefully presentations at the spring meeting. We also want to try to promote research to address our critical uncertainties, like try to see if we can maybe put some graduate students on some of these critical uncertainties. And we do hope to start crafting guidelines, facilitate use of ARUs in Cuckoo research and monitoring. And we were thinking, maybe we can start thinking about where can we use ARUs and what studies can we use ARUs? Maybe we can use ARUs to monitor CSWIG occupied sites. Okay, so that is the end of my summary. And I'm just going to look at
I know Tracy had her hand up at one point, and there's definitely questions in the chat.
Okay, yeah, so I just want to look and see. So yeah, we definitely have time here for some questions. So I'll try to answer your questions as best I can. Jenna, will you read me some of the chat questions, please?
Sure. Tracy, do you want to go first since you had your hand raised? Just so I don't.
Yeah, the only reason I was chiming in is because it looked like there was a pretty important question in the chat in the beginning about just the process and what an ARU was. But that was all.
Okay. Can you explain a little more about ARUs? How do they work, etc.?
Okay, so and anyone else who's on this call, like Dawn or others, can also chime in. But basically, when you think of a camera trap, right? When you put out a camera there and it senses movement and it takes a picture of an animal that passes through, passes by the camera. This is sort of similar. An ARU is a unit that you put out there, and it is actually recording the sounds that are out there and the soundscape in an area. And folks are using those units to see if they can survey for or detect cuckoos without actually having to go out and do the surveys themselves. And so it can be more cost effective, time effective to just have these units out there running for 3 days, 10 days, a month, a field season, and just constantly, running. And then you go out at the end of that time period, and you just bring them into the lab, and then you start processing the data. Now you're going to have a lot of time at the end processing the data, and not as much time, in the field. So there's a little bit of sort of balancing that out. But does that answer folks' question about ARUs? Yes. Okay. Thank you. Okay. Is there another question, Jenna?
Yeah, Linnea Hall just had a comment. I think deployment distances depend on how big the area is that you're trying to search with ARUs, as well as your objective. And I want to chime in also noises. Great,
that's a good point. I'll make a note of that.
Oh, Steve Albert asked, is 500 meters the currently accepted stop distance during protocol surveys, and 300 meters from the estimated cuckoo detection, or 100 meters between survey points. But you were saying 400 to 500 meters is the currently accepted, or sorry, is what people were saying their distances were for the ARUs.
Yeah, they were saying that, and they were saying that they were trying to sort of consider what the recommendations are in the survey protocol and try to somewhat follow that. So that it's like a surveyor is there listening for vocalizations. It's sort of taking the place of a surveyor.
Right, gets expensive with those more pricey units. For insects, is that just cicadas?
You know, that's a
Oh,
good question.
Nick
think-
answered that. I'm sorry.
What's that? Go ahead.
Nick answered that. He said
Oh, good.
cicadas, orthopterans, et cetera, can be picked up by ARUs with commonly used sampling rates, 24 kilohertz.
Good, thank you. Yes, I was going to say something to that effect. Thank you, Nick.
Linnea Hall had another comment. If you build your own system like we did, you don't have to wait as long to get the hardware.
Right, yeah. I was trying to touch upon that a little bit.
Okay.
Of course, you have to know
Claire-
how to do that.
Claire asks, given the challenges the ARUs face right now, is it plausible to survey for yellow-billed cuckoo without combining that effort with on-the-ground observers? And Dana answered yes.
Thanks, folks, for answering the questions also. in
Do
the
you think
chat.
so, Hira? Do you agree?
I
Hey,
think
that was not in response to that. It was in response to an earlier question. Sorry.
Oh, okay.
I did not answer that.
The overlap of the chat. Sorry.
yeah.
Thank you for
So,
clarifying.
so personally, you're asking me, Jenna, do I think that you can just put out the ARUs and without doing the protocol surveys? And I think that it really depends on your question. If you are just asking, are there cuckoos out there? Is there even a cuckoo in this area? Then I think you can do it without doing surveys. But if you want to answer more specific questions, how many cuckoos are out there? What's the breeding status? Right now, and it depends on kind of where your study is taking place, your study location. But I think it's really, I do think right now it's important to have some additional survey work in there to sort of QC and complement your ARUs.
Okay, thanks. Claire asks, has anyone written a protocol on deployment of ARUs for use of surveying for yellow-billed cuckoo?
No, that's what I was saying in the talk, was that we hope to be doing that. We're trying to work on that right now, and specifically Nick is working on that, Nick Beauregard. But there was that link that I had in my presentation, and we can put that in the chat later, or send it out to folks for the KITS lab. And I guess that there's some really good resources there. And Jenna just put it in the chat about how to use ARUs in general.
Okay, thanks. Let's see. Krista says Texas Parks and Wildlife just funded a project that used ARUs in the 2021 breeding season. Yellow-billed cuckoo detected at every deployment site in West Texas. I'm not sure how to say last name. Bowl et al. 2022 final report to the Texas Parks and Wildlife Department.
Great, thank you for sharing that information. And if you want to be, I should mention, I guess, if you want to be part of this email group, just send me an email and let me know. Or we'll be sharing some of this information and reviewing some of the documents, like the draft guidelines for use of ARUs in cuckoo research. And Jenna put my contact information in the chat. Thanks, Jenna.
And Nick also chimed in to Claire's question. We're working on a brief write-up to distribute for using ARUs for yellow-billed cuckoo. I'm also working on a manuscript for using Kaleidoscope Pro to process data for yellow-billed cuckoo. Ooh, that'll be really helpful. Thanks, Nick.
Okay, great. Thank you. Now, I want to try to stay on schedule as much as possible this morning so folks can log in and off to different talks that they're interested in. And we're not scheduled to start our morning session for another 3 minutes or so. So I'm just gonna let folks hang out here, or you can step away real quick. But we can also address any other questions that people might have during this little period here.
Hey, here, I don't
See
know if you're
some-
looking
Yeah,
at the
yeah.
chat. Okay.
Yeah, I was just going to say, yeah, Anna, I did get an email about your work and in Montana, and you are on our email list, and hopefully we can start connecting that way too. In terms of the slides being shared, I can make a PDF of this presentation and upload it onto our website. I actually have a lot of this information in a Word document, and that's the document that I wanted to just send out to some of the participants to review, to make sure that all of my information in there was accurate and, you know, was correct in terms of how I summarized their presentations. And then I'm going to take that and put that on our website. So I was hoping to put that Word document on there, but I can, in the meantime, just put this presentation, a PDF of this presentation, up on our website under the meeting information. So as I was saying, you know, when I do put this presentation up on the website, it's still somewhat in draft form, because I really want to make sure that the participants of that meet and greet have a chance to review what I wrote and have a chance to tell me whether or not it's completely accurate, or if it's missing something, if it needs to be revised. So just know that there might be some things in there that need to be tweaked a little bit, because I'm already getting, like, for example, input from Linnea about some of their work and clarifying some of their work, the results of their work. Okay, so let's see here. I just want to I'm going to start setting up for the morning session here. 1 second. Okay, so Katie Holland is going to be our first presenter today, and I just wanted to check. Katie, are you ready to share your screen?
I am. Would you like me to do that now?
Yeah, I think we're still going to hold out just a little bit more
Sure.
until we officially start, in
Yep.
case people are just tuning in for your presentation right now.
Okay.
And then we'll be on track, I think, with all the talks after the steering committee meeting. We kind of ended a little bit earlier than scheduled.
Sure.
Okay, great. I see your presentation here. Perfect. Okay, for folks that are just joining us, we just finished up our fall steering committee meeting, and we are going to begin the more formal presentations of the morning. And our first presenter is going to be Katie Holland, and she's going to talk about some of the road to recovery process, co-production, and social science. And I'm going to hand it over to her. Oh, there you go, Katie.
Yeah. Can you hear me okay?
Yeah, you sound perfect on my end.
Great. Thank you. Nice to meet everybody. Again, my name is Katie Holland. I am with Road to Recovery as a social scientist. Also, I'm a research scientist at Virginia Tech, where I serve as the National Bird Conservation Social Science Coordinator. So today I'm going to focus on the Road to Recovery process. As you all, I'm sure, have heard, Road to Recovery is very eager to support your working group. The yellow-billed cuckoo has been identified as one of our 4 pilot species. And so we're very invested in all of the work that you're doing together as a working group, and also individually as researchers and practitioners working on these important conservation efforts. So I'm going to talk today about the Road to Recovery process, the idea and concept of co-production, and why that's so important and fundamental to Road to Recovery. And as I do that, I am a social scientist, and so I'll be focusing on ways to incorporate human dimensions and social science research into those steps in the Road to Recovery process. I am open to questions anytime during the presentation. We'll have some time when I'm finished as well. I do my best to monitor the chat, but Hira and Jenna, if you see questions that you feel like are best answered in the moment, please feel free to interrupt me with those. So first, to start, what is Road to Recovery? We often refer to Road to Recovery as R2R. And Paul Schmidt put in the chat earlier, we do have a new website, which I'm hoping to have time to show you briefly at the end of my talk today, but it's r2rbirds.org. You can access it and explore some of the work that we're doing and who's involved on that website. We'll also post updates, news, and opportunities through that website as well moving forward. So Road to Recovery is an independently funded enterprise that focuses on the recovery of steeply declining birds in North America. The energy that led to the formation and the ongoing momentum within Road to Recovery came from the science publication in 2009 that documented the net loss of 3 billion birds since the 1970s. From that publication, a lot of energy across the bird conservation community was really sparked, and Road to Recovery is a product of that. We have a specific Road to Recovery process, which is what I'll be talking about today, that's specifically built on integrating biological and social sciences to co-produce research and conservation strategies targeted at these species that are in the most need of critical recovery. So Road to Recovery, we have 3 primary goals that we focus on. To move my chat box here. First, the development of a process for management, both management and recovery of birds. And I'm going to distinguish between those two here in a minute. Promote targeted and actionable science from both the biological and social sides to support recovery, and then to increase efficiency and effectiveness of implementation. How do we monitor our programs and our efforts to recover bird populations? How do we determine if we're seeing success, again, from both biological and social perspectives? So there's a big difference that we see within Road to Recovery between management and recovery. And obviously, with it being part of our name, our focus is truly on recovery of species. Management can be interpreted more broadly to look at wider ecosystem projects, efforts that are designed to support, let's say, a protected area that has been established or some sort of already determined intervention that's in place. Recovery, on the other hand, we view as more targeted. Recovery, the term recovery has traditionally been used in species conservation for both birds and other species for those that are already listed as threatened or as endangered. Road to Recovery as a group supports using that process more broadly to target bird populations and bird species who are not already listed, but their population trends show that as being a true possibility if targeted action is not implemented and soon. So to recover these species, we need intentional focus on the individual species themselves. Even bird species that use the same habitats for part of their life cycle may have different areas within that life cycle or geographically that are more important to the survival of those species. We as conservationists need to know where those targets are and for each of the individual species. Because again, even if they need the same habitat, or even if there are the same broader threats that are impacting those species, what we decide to do about them and where that action should be targeted may greatly vary across even similar species. So we're focusing on that recovery concept and targeting individual species recovery, specifically those who have been identified as experiencing these extremely steep declines. So what's our approach to doing this within Road to Recovery? We're doing several different things. So like I said, we're focusing on highest priority species. The State of the Birds report that was released just recently includes these lists, about 90 species identified as on alert and about 70 as tipping point species, meaning that they're in all of those species are in dire need of conservation and of targeted recovery, some more urgently than others, but all within the urgent category. So if you access our website, that list of species is available there. And as I said, it's also available within the newly published State of the Birds report. Once we identify those species, which we have, we want to know what are the knowledge gaps for determining population limiting factors. Those gaps exist within the biological sciences. They also exist within the social sciences. So we first need to know what the gaps are if we're going to try to fill them. How do we get data on those gaps and on these species collected in the most efficient way? What's the data that we need the most? Can we prioritize different types of projects for different species in need of recovery? Then we want to advance the species through measurable and actionable steps, and we want to monitor those steps as we move forward to ultimately produce a recovery plan and move this species along the road of recovery and see those measurable changes in species populations. So bird conservation is a human endeavor, and I would argue very strongly that conservation in general, regardless of what we're talking about, bird species, large mammals, the oceans in general, it's a human endeavor. And I spent a lot of time teaching undergraduate environmental science students, and I taught the human dimensions course for those students. And I always started this semester by asking, why do we need to understand human dimensions? The answer overwhelmingly every year was that, well, humans cause all of these environmental problems. True, right? Humans can be blamed for just about every environmental problem that we can think of. But that's not the end of the human dimensions story. That's a piece of it. Humans do cause problems. Humans are also greatly impacted by problems. Different groups of people are impacted differently by different environmental problems. And humans are also the solution to environmental problems. You don't make conservation progress without engaging people. And so however we want to view it, whether it's we have to engage people to move forward, or we want to engage people because we understand the impacts of these conservation goals to humans as well, it doesn't necessarily matter. What matters is that we all agree that human dimensions and social science incorporation is essential to recovering bird populations. And Road to Recovery does believe that. And our focus is on a co-production of research that incorporates both the human dimensions and the biological sciences. So as I've had conversations in this role, and my role more broadly as National Bird Conservation Social Science Coordinator, much of what I talk about is, what are human dimensions? What does that term mean? And what is conservation social science? Because a lot of times those are used interchangeably, and they're not necessarily the same thing. So before we move forward into the Road to Recovery steps, I'll clarify the difference that I'm talking about between those things. So human dimensions is a term, it's a broad umbrella term that encompasses a spectrum of both research and engagement strategies that focus on human behaviors. They also focus on the social impacts of conservation efforts. So you can see in this diagram here, some of the ways that human dimensions research and engagement can impact and be used in conservation programs. I'm not going to go into it in great detail, but I'm happy to follow up with any of these. I'm going to tell you at the end kind of my role and how I can support some of the work that you're doing. But it's certainly in doing some of this research and advising on some of these research methodologies that relate to understanding human behavior, and also in how do you use social science research that already exists to plan your engagement strategies. So maybe you don't have the capacity to be gathering new social science information. That's okay. A lot of really wonderful, powerful, well-researched social science concepts exist in the literature related to a lot of the issues that would affect the work that you do as bird conservation scientists and practitioners. So how do you leverage that social science research that exists? How do you use it to inform engagement strategies? And we do those things by using the basis of conservation social science. And so conservation social science is a term that refers to so many disciplines that evaluate these human dimensions problems. And so I have some of them in this diagram here, things like political science, communication, education, recreation is a big one for conservation that we don't think of all the time, psychology, economics, all of these are considered the conservation social sciences. Just as one biologist doesn't do everything under the umbrella of biology, one social scientist does not do everything within the umbrella of conservation social science. So it's important to acknowledge that and understand that, and then think about, as you recruit social scientists to work on projects, what is it specifically that you need? And that's something that I'm always available to help figure out as well. But we have to be targeted in what type of social science and what types of social science methods we're engaging in our projects, so that we're getting the most useful and most valuable data moving forward. So this brings me to our Road to Recovery process. And I'm going to go through these 4 phases that we've split the Road to Recovery process into. For each of the 4 phases, Road to Recovery really emphasizes focusing on species biology science and also focusing on social science. What type of social science and what type of biological science is incorporated in each phase may vary, but it's important to be thinking about those 2 columns as you move through all of the phases of the recovery process. For my presentation today, I'm focusing primarily on the social sciences, but I'll keep reminding myself and all of us that these ideas are intended to be paired with biological research as well. So starting with phase 1, assessing and learning, and again, looking at both social science and biological science. The first step is to identify the species that we need to be assessing and learning about, and we've done that. So in that State of the Birds report on our Road to Recovery website, the list of species that we have identified as needing immediate recovery efforts, all urgent. You could argue varying levels of urgency, but all considered urgent. We've done that, and we're focusing on our efforts on those species specifically. So then we have to work to fill in biological and social science knowledge gaps. I have all these pictures with different clicks. There we go. So this is a process that can be done a lot of different ways, and your working group specifically has a lot of amazing information put together on the yellow-billed cuckoo. This can be done through meta-analysis processes for the social sciences. So if there are identified social science issues that have come up that many of you have identified as impacting your work, there may be meta-analyses out there on those social science processes. If there's not, that's something that can be done, strategic ways of gathering and organizing social science information. For someone who's not in the social science field, it can be overwhelming because there are all these subdisciplines. But once we have identified the gap that you're looking to fill, we can identify what disciplines knowledge would really contribute to that gap and start pulling it together. Once we see that's out there, then we know what our gaps are, both biologically related to the species and from a social science perspective related to the relevant threats for that particular species. And this is where co-production really comes in. So once we know any information from either side, so if you are making progress with biological data and information related to the species, having that data can help to identify gaps on the social science side and vice versa. So co-production is really important at any stage in our projects, but ideally at the beginning. And so we want to bring in as many partners from as many different disciplines into the conversations that we're having to inform the decisions that we're making and to help us identify those knowledge gaps. So as some examples about what we might be looking for in this phase, from a biological side, we're identifying linked populations, migratory connectivity, vital rates, looking at full annual cycle models and trying to identify where in that cycle birds may be most at risk or where threats may be impacting the overall population at a disproportionate rate. From the social cultural perspective, some things to get your started on thinking about what these gaps might look like, understanding the capacity in an area for conservation. What is the social and cultural context of an area that is in need of conservation effort? How can we measure that? And how can we understand if conservation projects could even take off in that area, or if there's other social needs, cultural needs at play before a community may have the capacity to engage in conservation? Cultural relevance of a species. This is important for both protecting the species, but also as we move to those engagement pieces within the human dimensions umbrella, you have to understand the cultural relevance of a species. Some have very high cultural relevance, and so messaging needs to take that into consideration. Others don't have a cultural relevance for a particular community. And when that's the case, using that species to gather momentum or to energize a community may not be effective if there's no cultural connection to that species itself. And then feasibility of management options. Oftentimes when we identify threats to populations, there are various management options that could help. So understanding the feasibility of those in different parts of a species range is really important. Those are just a few examples, but ultimately they lead us to answering the question on the bottom of this screen in yellow: where does our work have the potential to be the most impactful for species recovery? We first need to know that, or not first, but we do need to know that from both of these sides. Biologically, within the species life cycle, within their migratory range, where are the areas that conservation is most needed and would have the biggest impact on recovery. And then we have to look at those areas from a sociocultural perspective as well, and understand if that type of conservation is feasible there. If so, that's great. If not, where in that range can you find kind of the sweet spot where you have social suitability and management options are feasible, and they're also identified as being areas that would be biologically very productive from a recovery standpoint for the species. Not a small task to get through phase one. There's a lot there. And I'll talk a little bit about moving through these phases at the end altogether. But this brings us to phase two, which is planning and design. So once we identify those knowledge gaps and we can work to fill them, once we answer some of the questions that we've identified, we can move on to this planning phase. And we're looking at limiting factors. And so, for example, if you identify limiting factors such as habitat loss and non-breeding grounds for a species, we can then use concept maps and we can link to social science concepts. So is that habitat loss coming from land conversion? Is it coming from agriculture? Is it coming from urbanization? Then once we know those things, we can think about what are the drivers for those changes? What are possible alternatives? What's the political government context of those actions that are leading to the habitat loss? We can start to develop webs of these human dimensions questions that then can lead us into being able to plan where we can target actions for that conservation effort. And we'd like to point out, ideally, we have this information across the species range and throughout the full annual cycle of the species. With working group conversations that I've had, this always comes as a barrier because for many species, we don't know all of this information yet. And this shouldn't be a holdup. So moving through these phases, it doesn't mean that we have to have all of phase one completed for everything related to that species. We can kind of move through the phases for different pieces of the puzzle of recovery of a species. But it's important to point out that the ultimate goal is filling in these knowledge gaps and having plans across all of these different pieces and phases. But recovery work can begin now, and it can begin at any of these points, wherever we have the capacity to be doing that work. And sometimes this leads us backwards in the road to recovery phases. So sometimes we get to designing, we have great questions, we've made great progress, we have momentum and funding, and we're designing a research project. And in that project design process, we identify more places where we need to assess and learn, and we identify bigger knowledge gaps. And that's okay. And we can make strategic decisions of when to move backwards and when to continue moving forward. I think ideally we do both, but I know the reality is that that's not always an option. So phase three is piloting and implementing once we have the plans in place and we've designed plans, ideally through co-production, incorporating insights from both the social sciences and the biological sciences to make those plans that we can then pilot and implement. And I'll point your attention here for some examples. I tried to pick a few examples to present today, but I thought for the sake of time, I probably wouldn't get into the specifics of them. But on the NABC Human Dimensions website, we have a great bank of social science human dimensions success stories related to bird conservation. And at some point when you have time, it's worth a scroll through those stories. They're all very short, but I think they do a great job of sparking ideas of what on the ground social science implementation can look like to support bird population recovery. And then finally, phase four is monitoring and evaluation. And this is one of the questions as a social scientist I get asked a lot, because oftentimes there are some community engagement projects in place, or there are some human interventions in place to change human behavior related to a conservation project. How do we monitor those things? So I will leave it to most of you to explain how you best monitor the bird populations, because again, I am a social scientist. But how do we monitor conservation actions? It's very complex. And one of the things that's first important to do is think about, are implemented strategies successful? But within that question, there's a lot that can be monitored. And the first most important thing that I want to emphasize is that we have to be on the same page with how we're measuring success. Oftentimes, as conservationists, we view success by population estimates of the species. And in bird recovery, which is the goal of Road to Recovery, that will always be very important. Are we seeing changes in the bird populations over time? Are we recovering populations? But just as within biological monitoring, you would need to break that down to have more frequent indicators, we have to do that from a social science perspective as well. So we can do that by measuring participation in different conservation strategies. We can measure attitudes towards those strategies. We can measure impacts on communities from a social perspective in several different ways. And then that answers the question of how do we know we're being successful? Sometimes there may be a lag before we see actual biological indicators of success, but we may have early indicators of success or lack of success from the social science perspective. So it can be really beneficial to biologists to implement some monitoring of the human dimensions and of the conservation strategies in place from a social science side as well for that interim data. Are these conservation strategies successful from a social perspective? Are they sustainable? We use the term sustainability a lot in environmental science, but it applies to all sorts of social science as well. Communities change, individual people change. So are targeted interventions that are put in place sustainable over a period of time at both that individual level and then all the way up to more of a societal level? Are the interventions sensitive in considering the social and cultural context? Has local input been used in developing these strategies? And is there a pulse on that cultural context throughout any changes that are made to the intervention strategies? How do strategies impact human well-being? There's a huge body of research on how conservation efforts impact human well-being. Are we keeping track of that? And is human behavioral change occurring? If it is, do we understand why? And are we able to leverage that why to the benefit of the species to move them towards a recovery goal? Those are just a few examples, and that's something that I'm here to assist with that monitoring process as well. So coming back to looking at the Road to Recovery phases all together, like I said briefly in that previous slide, ideally we have all the knowledge that we need. We fill in all the knowledge gaps from both the species biology science and the social science perspectives at the beginning, and then we have all the information we need to move forward through these phases. I think we all know that that does not happen, and recovery of bird populations cannot wait. So we have to break apart our efforts as a collective group into the work that all of you are doing individually. And can we advance that work through these phases while keeping a pulse on overall, how are we doing as a community for yellow-billed cuckoo conservation? I know that many of you are actively engaged in a lot of the biological science and monitoring. I'm here as the Road to Recovery social science scientist to provide some social science support as well. So what do I do in my role? These are just some of the things that I may be able to help you particularly with as you work on yellow-billed cuckoo conservation. So consulting on projects, advising on how to truly start the co-production process and what are some strategies to identify partners who may contribute to that co-production concept. Development of appropriate social science research questions, reviewing data collection tools, or helping with analyzing your social science data that you collect. I spoke specifically about those monitoring plans. That's something that I can help to create. Incorporating human dimensions or conservation social science goals and components into grant proposals that you're writing. Funding is often a barrier, so I encourage you in any grant proposal that you're moving forward with from here on out to consider incorporating a social science goal into that proposal to be able to have some funding to get this social science piece going and support co-production in your own work. And then helping to make social science connections, accessing relevant research, pointing to different sources of information within the social science community. And as Road to Recovery grows and evolves and gains momentum, my position does as well. And so it will continue to evolve, but a primary part of my job currently with Road to Recovery is supporting the 4 pilot species working groups and you are one of those. So I'm available, as Paul stated in the chat earlier, Road to Recovery as a whole is very eager and excited to support your working group. So we're here all the time. We want to get to know members of the working groups and know the work that you're doing, and support you in any way possible. So this is my contact information, and I'm going to put it up here in a minute. I think if I have one more minute, I'm going to try to share, where did it go? I guess I need to do this first. There we go. I'm going to try to share the Road to Recovery website just quickly with you. Oh, it's not letting me switch my screen share. Let me stop first, if that's okay. And it's in the link as well. Thank you. I don't know why it won't allow me to switch here, but I'm going to try it one more time. And if it fails, I will abandon ship. Here it is. Okay. So here's our Road to Recovery website. Explore it whenever you have the time. But just as a quick plug, if you go to staff and committees here on our website, you'll see my face as a social scientist and a button here to schedule a consult with me. And this would allow you to just go ahead and schedule a meeting onto my calendar. I would love to talk to you. I'd love to hear about the work that you're doing, ways that you're already incorporating social science. I'm always looking for positive examples of social science incorporation into bird conservation that I can help with, or talk about ways that you can start doing that if that's not already included in your work. So I will stop there, and I'm happy to answer any questions that may have come up.
Thank you so much, Katie. So we have 2 minutes for questions. Are there any questions for Katie? Do people want to raise their hand or put their questions in the chat?
I'm going to put my email in the chat too, because I did talk longer than planned, and any follow-up questions can be sent to me.
Emma Garcia, I see that you have your hand up. You can unmute and ask your question.
Yeah, hi, nice to meet you. I'm Emma Garcia. I live in Los Cabos. I'm in charge of the Conservation and Key Species Laboratory here in the Autonomous University in Baja California Sur in Cabo San Lucas. And we're working hard with the yellow-billed cuckoo since 4 years ago. More than a question, it's like one thing that we're focusing, it's in social science conservation, because here is a strongly, well, cuckoo is a strongly cultural connotations everywhere, but here it's really, really, really strong. Here it's called pájaro chubasquero, that it's the rancher's name for rainbird.
Okay.
And that's a thing that we're doing because here the monitoring is really hard because of the landscape. And we're working with the ranchers and the ejidos to do the monitoring. And next year, we're going to do the first festival of the yellow-billed cuckoo here in Baja California Sur. And it's going to be in Caduaño. We're working on that right now with the government. And we would love to work with you. And we're working strong with that. So, and also with everyone that wants to work with us, because we really need help. So more than a question, it's like I'm eager to participate with you and work with you, collaborate.
Likewise, that sounds amazing. Yeah, please reach out. I'd love to discuss that further. It sounds like an amazing opportunity.
I don't know if I can share the screen quick so I can show you just a quick slideshow.
Well, right now we have to move on to the next talk.
Okay, I will send you the slide of the festival for all of you guys.
That would be great. You can also send it directly to the listserv, the same address that we send out to everyone else. And I know that Jenna had put that in the chat, and that will go to everyone. So you can just you can actually use the listserv to send out announcements. It's not just from the working group to the members. The members can use it as well.
I will do.
Okay, great. That's wonderful. That's really great to hear. I'm excited about that.
I just put it in the chat.
Okay, thank you, Jenna. Okay, thank you, Katie. So now we're going to move on to our next talk by Clark Rushing. And I'm just going to see if, Clark, are you available? There you are.
Yep,
Hi, you ready to share your screen?
Yep.
Great.
So I'm going to talk a little bit today about structured decision-making, or at least a portion of the structured decision-making process that is being used in a couple of different contexts for different species working groups to try to prioritize research and monitoring needs to aid management. And I wanted to just very upfront acknowledge that everything I'm going to talk about today is the products of several collaborations. So a lot of this work started with a collaboration with some colleagues at USGS, Mike Runge and Jim Lyons, who are at the Patuxent Wildlife Research Center, and Madeline Rubenstein, who is at the National Climate Center with USGS. And the kind of initial stages of this value of information approach came from some work we did a few years ago that was published in Biological Reviews, and I'll go over that today. We sort of started this approach. It's been used in a number of contexts and refined and improved by some other colleagues, particularly Abby Lawson, who was at USGS Patuxent, is now the assistant unit leader at the New Mexico Co-op unit, so still with USGS. Another USGS postdoc, Michelle Stanchel, who's now with the National Park Service in California, and Janice Gardner, who's a researcher at the Sageland Collaborative in Utah. They're very instrumental in sort of taking the ideas that Mike and Jim and Madeline and I came up with and really refining them in a few other case studies that I'm going to highlight today during this talk. So just to kind of give a little bit of context for where some of these ideas came from and what we're trying to think about. One of the challenges to migratory bird conservation, as I'm sure all of you know, is that the environments that these species inhabit is changing very rapidly. It's changing for a variety of different reasons: land use change, climate change, all sorts of threats, and for migratory species, those threats are happening across their full annual cycle. And so there's a lot, it's very challenging to think about how we make management decisions in these contexts, partly because there are so many different threats that these species face. And as I'll highlight throughout the talk today, also because we have a lot of uncertainty about where those threats are, how these systems work, and how species will respond to our management actions. But we have to make decisions in the face of all of that complexity and uncertainty. And the way that we try to do that oftentimes is through the process of structured decision making, which is a sort of a form of decision analysis that has been widely applied in natural resource management. And I'm not going to give sort of a full overview of the structured decision-making process today. Many of you are probably familiar with this process and this figure in particular, showing this PROACT cycle that's used to make decisions, where we start by framing the problem that we're trying to address. Then we set measurable objectives that we can use to determine if our decisions are successful or not. We articulate different alternative decisions that we're trying to choose from. We then try to predict the consequences of those different alternatives in terms of how the system will respond, and use those predictions to identify trade-offs and hopefully make decisions that will have the biggest impact on meeting our objectives. So that's sort of the ideal case for how structured decision-making works. It's obviously a very difficult process. And one of the things that oftentimes really bogs down efforts to do structured decision-making is that there's a lot of uncertainty. So when we think about species like yellow-billed cuckoo, I think we could all outline a lot of different sources of uncertainty that we have about where this species is limiting, what the threats are to the species, whether those are on the breeding ground, the wintering ground, during migration, what demographic rates are the cause of the decline. We just have lots of different things that we don't know about systems like this. And we also don't know how the systems will respond to our management actions. So a lot of this uncertainty sort of comes at this stage in the structured decision-making process where we're trying to predict how our management, how our system, how the species will respond to management actions. And it's very easy oftentimes to sort of throw up our hands and say, well, there's so much uncertainty, we can't do this kind of modeling that we need to do to predict our management actions. And therefore, we usually sort of revert to like sort of heuristic methods for making decisions, when ideally we would still use this approach. And I want to walk through very quickly one concept that's useful for thinking about uncertainty and how it influences decision making, and that's what's called the value of information, or sometimes it's called the expected value of perfect information. I'm going to go through this very quickly. But if you want to know more, this paper by Mike Runge and Sarah Converse and Jim Lyons called "Which Uncertainty" is a really good kind of in-depth explanation of this concept of value of information. But to kind of get to the way we've been using this approach, I do want to walk through, at least conceptually, how value of information works. So I'm going to use this figure to kind of make this point. And this is a very simplified case where we have two hypotheses about the way our system works and about the way it's going to respond to management. So we'll call those hypothesis A and hypothesis B. And to use this value of information, we also need some metric of how strongly we think hypothesis A is true versus hypothesis B is true. So we'll label this x-axis sort of degree of belief in hypothesis B, and that would go from 0 if we knew for sure that hypothesis A was true, all the way to 1 if we knew for sure that hypothesis B was true. And then we imagine that there's some management action that we could take. And oh, sorry, I forgot to label it. So we'll just call our degree of belief P star here. Doesn't matter where it is on the x-axis here, but we'll say this is before we do our research or monitoring or make our decision, how much our degree of belief in hypothesis B. And so we can sort of imagine this scenario where we have a management action, maybe this is like restoring breeding habitat or something like that. And if hypothesis A is true, where this line intersects with the y-axis is the benefit to our system, whereas if hypothesis B is true, then we get the benefit up here at the top. And the key here is that before we make this decision, we don't necessarily know which of these two hypotheses is true, but we can think about the expected benefit as being sort of the average of these two at the point where our degree of belief intersects that line. So if we don't know which one is true, we take the action, and then later we figure out hypothesis 1 is true, or A versus B, then the benefit that we can expect in the face of that uncertainty is just going to be the average of those two outcomes weighted by our degree of belief, and we'll call that point A. In this case, if we only had one action that we were trying to decide to take, there's no decision to be made. We just take action A because that's the only one that we have available. So there's not really a decision to be made in this case. But now we can imagine that there's a second action. We'll call it action 2, and you can see that if hypothesis A is true, we do better if we take action 2. If hypothesis B is true, then we do better by taking action 1. But again, we don't know which of these hypotheses is true before we make the decision. And so in this case, if we could sort of predict these outcomes under each hypothesis, then we would say, well, in the face of this uncertainty, the kind of average expected outcome for action 2 is higher than action 1. So we should take action 2 in this case because we do a little bit better on average. So if we have to make the decision in the face of this uncertainty, we should choose action 2. The idea behind value of information is that instead of deciding which action to take when we're still uncertain about the hypothesis, we could learn more about the system and resolve that uncertainty and then choose what action to make. So in that case, if we did some research and we learned that hypothesis A was true, we would always take action 2. If we learned that hypothesis B was true, we would always take action 1. And if that was true, our expected benefit would be the average of those 2, which we'll call C. And in this case, because our expected benefit is higher by resolving the uncertainty than it would be if we made the decision in the face of the uncertain. When we were uncertain, we should do the research and we should figure out which hypothesis is true, and then take whichever action would have the better outcome. And so this is the idea of value of information, that by resolving the uncertainty, we improve the outcome of our management action, and therefore there's value to doing some research or monitoring that resolves the uncertainty. And just to kind of illustrate that is not always the case, we could imagine a scenario like this, where these are the outcome of our 2 actions, and it doesn't matter which hypothesis is true. Action 1 always outperforms action 2. And if this is the case, we still might want to learn about the system, but it doesn't change the action that we would take. So in a scenario like this, there is no value to resolving this uncertainty in terms of making the decision, and therefore we should just choose action 1 and not spend time and resources doing the research. So it's not always intuitive ahead of time which uncertainty we should try to resolve versus just making the decisions. So this is the idea behind value of information. In that Runge paper that I mentioned a minute ago, they talk about a related approach called the expected value of partial information, which involves multiple sources of uncertainty. And that's sort of the gold standard approach for prioritizing research needs. Which source of uncertainty will benefit our management the most, and therefore we should invest resources in management. The problem is, as I mentioned before, it's very difficult to do this, because in order to estimate these things, we need to express our degree of belief in these hypotheses in a probabilistic way, which is difficult to do. And we need to have models that allow us to predict the outcome, the expected benefit under alternative actions. And as many of you probably know, that is also very hard to do, especially for species like yellow-billed cuckoo, where we lack even basic information about demography, about limitation, about migration, and those types of things. So value of information is oftentimes difficult, if not impossible, to implement in these cases where we have a lot of uncertainty and very complex systems. That's sort of where the initial part of this project started, which was trying to come up with a way to do this value of information method in cases where we have a lot of uncertainty. And so we came up with what we're calling right now qualitative value of information, which is an alternative to the quantitative value of information I just outlined, that still provides a rigorous and transparent way to prioritize research needs when this quantitative value of information is not possible. The way this idea works, I'm going to kind of walk through very conceptually how this qualitative value of information works. And there's a paper that's in revision right now. Hopefully it'll be out soon, showing that this qualitative value of information is theoretically a proxy for quantitative value of information. And what we do is we take the value of information and we divide it up into 2 components. The first one is called the relevance to the decision maker. We'll call this relevance for short, which sort of measures, again, how much would resolving this uncertainty change the decision and benefit the system relative to sort of taking no action. I'll talk about these scores in just a second. This is just sort of a heuristic way we in this paper we outline thinking about relevance to the decision maker. We also sort of break up quantitative value information into what we call the degree of uncertainty. So this is in the current state of affairs, how uncertain are we in this hypothesis? And so we can sort of think about our degree of uncertainty now as being on this kind of numerical scale. So we have these two metrics, relevance and uncertainty, that it turns out are a proxy for value of information. And the way this approach works is that we basically score each of our hypotheses using a standardized rubric, and usually some combination of expert judgment and expert elicitation. So I'll walk through the way this approach works in a little bit more detail in just a second, but we come up with a rubric that allows us to sort of score our different sources of uncertainty from 0 to 4 in terms of how relevant that source of uncertainty is to the decision makers, and how uncertain we are in the hypothesis currently. You'll see there's a third column in this table, in this rubric that's, oh, sorry. So just to back up for a second. So we score each source of uncertainty in terms of relevance, uncertainty, and then we multiply those scores, and that is the qualitative value of information. So it can go from 0 all the way up to, in theory, 16, or excuse me, 12, because relevance only goes up to 3, and that's our qualitative value of information score. The higher the score, the more value there is to reducing that uncertainty in terms of expected management outcomes. We have a third category that we've been using in these case studies that we call reducibility. This doesn't really have a direct corollary in traditional value of information analysis, but it's sort of measuring how easy is it to reduce this source of uncertainty. And again, we score that from 0 to 3. 0 being we could not reduce this uncertainty even if we wanted to, because we don't have the technology. It would take forever. It's way too expensive. To 3, which would be this is low-hanging fruit. If we just put a PhD student on it, we could reduce this uncertainty a lot. So each source of uncertainty gets scored in these 3 categories, and then we end up with a way to prioritize research. Just to kind of walk through how this generally works, I like to think of this as a 6-step process, and it's carried out through a combination of sort of workshops and expert elicitation. The first 3 steps are similar to any structured decision-making process. We can't think about value of information and decision-making outside the scope of the problem and the objectives. And so the first step to this is framing the problem, setting measurable objectives, and then identifying alternative management actions that we're trying to decide between. So again, these are usually done with workshops with different stakeholders, and I'll outline a couple of examples of where we've done that. So we go through these first initial stages, and then the next part, and what's really proven to be the most difficult part, is articulating the key sources of uncertainty that are hindering management decisions currently, and framing those as hypotheses that we can link back to management. So ideally, these are sort of the sources of uncertainty are framed in terms of a null hypothesis, like there's no effect of management on this process, versus there is, and we can link those hypotheses back to management actions. Again, I'll show some examples in just a second. And then we use this standardized rubric with expert elicitation to score those hypotheses. And that is usually an iterative process. So we usually, as we found, it's best to sort of score the hypotheses, talk about the scores, come back, allow people to revise their scores, and then end up with sort of the final scores for each hypothesis. And then the last step is sort of ranking or prioritizing those hypotheses. And because we have 2 measures, the qualitative value of information and the reducibility, it's best not to think of these as rankings, but as priorities. And just to illustrate sort of why that is, you can sort of imagine hypotheses that score very highly for both reducibility and value of information. So these would be hypotheses where we said they're very relevant to the decision. There's a high degree of uncertainty, and that uncertainty could be easily reduced through targeted research and management. So if all three of those scores are high, we have hypotheses that are important and that we can reduce, and we would usually say those are the highest priorities for investing research and monitoring into. Then we end up, and I should say, if we have kind of the opposite end, if we have low QVOI scores and low reducibility, those are probably sources of uncertainty that we should not prioritize, right? They're not very relevant. There's not a lot of uncertainty, and the uncertainty that is there would be hard to reduce. And so it's probably not, we're not going to get a lot of bang for our buck on those hypotheses. In these kind of upper left and lower right quadrants, we get into some trade-offs where we can imagine hypotheses that we've identified as being really high, really important. So their qualitative value of information score is very high. So we're saying there's a lot of uncertainty, and that uncertainty is relevant to the decision maker, but it's going to be difficult to reduce because the reducibility score is low. So you can think of those as like really important but difficult source of uncertainty. And then in the bottom right, we might have hypotheses that we've said, well, they're not super important, but we can reduce them quite easily. And so these might be sort of low-hanging fruit. They're maybe not going to get us as much value, but we can do something about it. And we've had some really interesting conversations in these working groups that have gone through this process about kind of how to prioritize those, which ones of those might be higher priorities. And I can talk a little bit about those discussions if there's questions about it. So that's kind of the end goal, is to rank hypotheses or prioritize hypotheses based off of these scores. And again, this is a rigorous process. It's transparent, and it gives us a way to sort of think about different sources of uncertainty and which ones are going to have the biggest impact on our management outcomes if we could reduce them. So that's kind of an overview of how this process works. I wanted to then spend the time I have left kind of going through a couple of case studies where this has been applied. I'll say this is a very new method. So this biological reviews paper that came out in 2020 was kind of the first attempt to use this method. Since then, there's been, that I know of, 3 other uses of this. So Abby Lawson and Michelle Stanchel, who I mentioned at the beginning, have gone through this process for black rails. So there's been some work to prioritize research on black rails, both on the Atlantic coast and the Gulf Coast using this method, and then rosy finches, which I'll talk about in just a second. So I'll sort of preface all this saying this method is evolving as we go through more case studies and figure out how it works and how to improve it. But this first time, the sort of initial use of this was some work that we did with the Fish and Wildlife Service, who the refuge program is tasked with buying parcels of land to protect migratory birds. The way that that prioritization currently works does not include any sort of climate change. So Fish and Wildlife Service was a little bit worried that maybe they need to change the way they decide which parcels of land to buy for the refuge program if climate change is going to impact migratory birds. So that sort of set the decision context for this. And then Madeline Rubenstein, who is with USGS, sort of took this on initially as a structured decision-making process and looped in Michael and Jim, and then I got involved as well. So the way we did this is Madeline first organized 2, I think there were 2, maybe 3 workshops that brought together researchers, decision makers, land acquisition folks to think about how climate change might impact migratory birds. So these were 1 or 2 day workshops with everybody sitting around brainstorming all the myriad ways climate change might impact migratory birds. We took the output from those workshops and sort of distilled all of that down into 11 hypotheses that we then grouped into sort of 4 broad categories. So we had hypotheses about how climate change is going to impact the distribution of birds, how it's going to impact the abundance of birds, how it's going to impact migration and phenology, and how it's going to impact community level interactions among different bird species. So using this kind of expert elicitation and then some judgment on our part, we tried to formulate all of this different uncertainty about climate change into these kind of 11 core hypotheses. We then created that scoring rubric for each of the 11 hypotheses, and we used sort of a combination of smaller group of experts, then with input from the Fish and Wildlife Service folks who work in the refuge program and know the way these land acquisition decisions work sort of inside and out. So again, it was an iterative process where the scoring was done by the experts. We then talked to the decision makers and they sort of said, well, that's actually not super relevant to us because of the way our decisions work, or that is really important, revised those scores a bit and ended up with a sort of final set of scores for those 11 hypotheses. If you want to know more about it, that biological reviews papers has all the details. This is those 11 hypotheses, kind of the way they ended up coming out of that scoring. I apologize for this. Note that now I have QVOI on the x-axis and reducibility on the y-axis, but we still have those kind of quadrats of importance, and these are color-coded by which of those 4 categories the hypothesis was in. And you can see the hypotheses that were identified as kind of the highest priority for research, for the most part, had to do with distribution. So uncertainty about how climate change is going to impact the distribution of migratory birds. And that intuitively makes some sense, since the decision is about land acquisition, which is permanent. And if the distribution of birds is changing over time, you risk making bad decisions about where to purchase land. And so that sort of intuition about which hypotheses were most important tended to be apparent in the way these hypotheses came out. We also, the questions about migration and phenology tended to be a little bit less important, which again, changes in the timing of how things are happening is a bit less important to land acquisition than distributional changes. So this is how we hope that the output of this kind of comes out. This can be then used by organizations like USGS and Fish and Wildlife Service to prioritize where they want to put research dollars in terms of climate change research on migratory birds. I'm going to go through this last example real quickly, if that's okay, Hira.
Well, we've run out of time for questions.
Okay.
But I know that the next speaker, I was just doing some texting here back and forth, is not going to probably take up that whole entire time slot. So we could just push it just a little bit further if this will be really quick.
I'll just go through it really quick and just say
great.
we did this for rosy finches as well, the 3 species, so slightly different context in terms of decision making. I'll skip ahead to we had these big groups, this rosy finch working group, where we identified the alternative management actions, and then we had everybody sort of in these breakout rooms on Zoom create these influence diagrams of sort of saying, okay, how does each of our management alternatives, how do we think it might impact our objectives that we set as a working group? So each of these lines is sort of a hypothesis, and then we use those to come up with our hypotheses about uncertainty regarding rosy finch responses to our management actions. So we came up with 9 hypotheses as a group. We scored those using the method that I mentioned earlier, sort of giving everybody in the working group a chance to score the hypotheses, a workshop where we went back over them, allowed people to revise their scores. And then this is sort of an interesting case. This is very new. This is within the last month or 2 that we've scored all these. And we ended up with this case where nothing came out as like super high priority. So we didn't have any hypotheses that we thought were both really important from a value of information perspective and really reducible. And so this group is sort of left in this middle ground of sort of saying, well, what do we want to do? Do we want to prioritize things that are easily reduced, but maybe a little bit less important, or hypotheses that we think are really important, but are going to be difficult to reduce? I can just leave it here. This was sort of my lessons learned, but I can just take a few questions if there's time, or happy to answer questions over chat or later.
Yeah, let's open up the floor to questions. Thank you so much, Clark. That was really interesting and just so relevant to the work that we're doing as a working group. I really appreciate you presenting this today. So are there any questions for Clark out there? Just raise your virtual hand or put it in the chat. I am going to change my participant. Sorry, was you, Valerie?
I'll say too, I think there's some Rosie Finch working group members that are also Yellow-billed Cuckoo working. I won't call them out by names, but they can answer questions about how they felt like the Rosie Finch process worked.
Am I missing any hands up? If I am, just those are two participants has raised hands, so I'm not seeing it with the screen share on right now. So let's see here. I apologize.
Russ has his hand raised.
Okay, thank you so much, Jenna. Okay, Russ, thank you. Please ask your question.
Hey, thanks, Clark. I'll try to lower my hand too. Having been through the Rosie Finch process too, I'm curious if there are sort of either pitfalls or some benefits from that approach that you can point this group towards, given that we are sort of in a similar place with maybe some broad, we have some broad areas. We don't know what the issues really are. It's hard to move towards a consensus view, and we really have a lot of options on the table. Is there a way we could have been more focused, for example, to get more value out of the process, or do you have a more definitive approach?
I don't know if I have an answer for how we could be more focused. I think some of the, as Russ is sort of alluding to, there was a lot of work that went into formulating these hypotheses. And one of my sort of lessons learned here is to have a really good moderator. I do think having somebody who understands this process and can keep the group focused on sort of the decision-making process and how the hypotheses link to the decisions is really important, because it's very easy to go down rabbit holes of, isn't this a really important source of uncertainty from a research perspective, even though it might not be related to management actions. And good moderators make a big difference. Yes, I will highlight what Russ just said, is that I will put in now that Abby's in New Mexico, you should probably rope her into this working group because she is an excellent moderator. So that was part of it. I think the other thing that we're sort of struggling a little bit with right now, which we're just going to have to go back to the drawing board and think about, is that we had some discussion. We got these kind of scores done, and we had some internal discussions amongst some of the working group members who were saying, well, actually, I don't think that should be a high priority because we can't do anything about trout stocking, or there's no way to restore native seed food sources in the Alpine, which points to me to a little bit of issue in the process, because in theory that should be reflected if they got ranked highly in the scoring, and then you're saying after the fact, well, no, we can't do anything about that. That should be reflected in the scores to begin with. And I think that reflects maybe a little bit of misunderstanding during the scoring process of what we wanted people to do. Despite, we had several rounds of scoring. We hosted like office hours, basically, where Abby and myself and Michelle and Janice sort of just sat on Zoom, and people could come in and ask questions. And so I feel like we worked really hard to avoid that. And yet there was just still some discrepancies between the way people scored and then after the fact what they told us about what they thought about those hypotheses. So but again, I think really good moderation, a really good moderator can help avoid those. And Abby's probably the best in the biz now, since she's done this for both black rails and rosy finches.
Okay, Clark, I think we're gonna have to stop there. Thank you so much. If
Yep.
we didn't address some of your questions, maybe put them in the chat, and if Clark sticks around, we might have some time after the next two talks as well.
Yep, I can answer a few in the chat while we're waiting.
Great, thank you so much, Clark. I appreciate that. Okay, our next speaker is John Stanek, and John, are you available to share your screen?
Yep, just a second.
Great, thanks, John.
Hi, everybody. Can you see my screen?
Yeah, it needs to be in, I think, slide view. Yeah, there you go.
All right. So a real quick update for you all for the Rangewide Cuckoo project. My name's John Stanek, and I'm just giving you the update, but this was a massive collaboration between nearly a dozen different wildlife agencies within the Western Cuckoo DPS. So this was a huge effort. Took a lot of coordination, a lot of state coordinators implementing this, basically following the Fish and Wildlife Service protocol. So this is a very large project, and I'm just here giving you a few details of what we found. So the Rangewide Cuckoo project had three primary objectives. So we wanted to develop a species distribution model for the cuckoo. So that was a two-part objective where we developed a preliminary species distribution model, and then we would use that for our sampling frame for our Objective 2 survey effort. And then following the data that we got in 2022, we would feed that back in and finalize an SDM for the yellow-billed cuckoo. That Objective 1 is being overseen by Utah Department of Natural Resources. And I think I just saw Russ Norvell on the call. And if you have questions, he would be the best one to answer those. Our second objective was to conduct a range-wide survey for yellow-billed cuckoos in 2022. And our proposal initially stated 250 sites, 250 sample units. We did this using the standard Fish and Wildlife Service protocol, with one small modification. To increase our sampling effort, we only conducted 3 surveys instead of the normal 4. So we did not conduct the regular last survey typically done in August. From historical observations, we know that that usually produces the fewest number of cuckoo detections. So we did not do a 4th survey at these sites. We only did three surveys at our sites. And our third objective is to evaluate the utility of ARU data. How is detectability by ARUs? How does that compare with our detection and occupancy estimates from surveys? So this third objective is being overseen by California Department of Fish and Wildlife, Neil Clipperton. I'm not sure if he's on the call or not. But we set out ARUs in California at 2 areas, in the Kern River Valley and the lower Colorado River at, I believe, Palo Verde Ecological Reserve. So 2 of the largest populations of cuckoos in California. And I've yet to hear about the results of those ARUs, but we had surveys at both of those areas. So we have survey data, ARU data, we have the spatial data of where the call broadcasts were played, we have the spatial data of where cuckoos were detected during those surveys. So it'll be pretty interesting to see that comparison there. And again, that will be analyzed by California Fish and Wildlife Service. So for the rest of this talk, I'm going to focus primarily on our second objective, conducting cuckoo surveys range-wide. So the proposal outlined 250 sites to be surveyed. We finagled the numbers and we upped that a little bit to 270 total sample sites was our desired goal. From Objective 1, we got a species distribution model of potential habitat. That was the foundation for our sampling frame. It's a collection of 1 kilometer by 1 kilometer grid cells of potential cuckoo habitat. Those grid cells were randomly ranked, And then we assessed those sample units in rank order. So effectively trying to pull out a random sample of the modeled habitat. We developed a rule set to include or reject sample units for survey. So we tried to make this as objective as possible to minimize our subjective desires of where to survey or what is or is not considered habitat. So some of these rules were logistic based. We needed to have enough time to access a survey to get in and get out. Well, specifically to get in the morning. Needed to have enough habitat to warrant the effort of conducting a survey there. And that was set at a minimum bar of 1 kilometer worth of habitat within and adjacent to the sample unit. So a minimum of 10 survey points was our desire for each and every sample unit. Safety was a consideration. We dropped sample units that were recommended to be possibly unsafe. And of course, landowner access and permission. We needed to have permission and access any of our sample units. This was a huge effort by state coordinators to go through all of our sample units in rank order and first identify landowners and then contact landowners. And it was a significant process, especially for Arizona. I believe Arizona needed written permission from every private and maybe public landowner to conduct surveys there. And lastly, well, 2 more points. As part of this project, we set up a goal of no duplicate survey effort. So if another entity was going to be conducting surveys at a sample unit in 2022, we let that other entity conduct those sample units. Specifically, the biggest partner that fell into this category was the Bureau of Reclamation on the Lower Colorado River and the Rio Grande. They have an extensive long-term Cuckoo survey monitoring project in both of those rivers. And where our sample units overlapped their areas of survey, we let BOR Reclamation conduct those surveys. Now, so we skipped over those sample units, and at the later on in the season, we hope to gather up data for those sample units and incorporate those into our future analyses. So in the end, we completed approximately 320 surveys at approximately 320 sample units, also referred to as study sites or sites. And we collected all of our data using field maps on personal smartphones. We did not get much, if any, pushback from our field staff, approximately 50 field technicians across the Western United States. We asked them to use their own phones, and they did so. They put field maps, ArcGIS's field maps on their phones, and collected data using a geodatabase that Utah helped develop and host. So here's a map on the right of what our final accepted sample unit distribution looks like. From the previous map, you can see that it was heavily weighted towards Arizona and New Mexico. And that initial distribution allocation of sample units was based in the proposal based on the proportion of critical habitat spread across the United States, the Western DPS. And so the final model, our proportion or allocation didn't vary too far from that in the end. And so you can see an example on the left of some of our 1 kilometer sample units. And these sample units were all surveyed as indicated by the blue survey points. And the black lines roughly indicate where the surveyor walked to complete these sample units. So in some areas, we confined our surveys to wholly within the sample unit, but that was not practical for most of our sample units. For most of our sample units, the survey passed through a sample unit and surveyed the suitable habitat within the sample unit and an extensive amount of suitable habitat adjacent to our sample unit. Now, a few things of note here is that we don't have many sample units that we surveyed on the lower Colorado River in between on the border of California and Arizona. A Bureau of Reclamation had that area covered extensively. And same with the middle Rio Grande in New Mexico. Bureau of Reclamation surveys most of the good habitat, most of the habitat south of Albuquerque or Belen down to the Texas border on the Rio Grande. So we did not survey that area in New Mexico as well. We relied upon Bureau of Reclamation to conduct those surveys. So here are results, just a coarse map of where we got cuckoos. And so the right-hand column of this table shows the sites or the sample units where surveys were conducted. And the left-hand column shows the number of total detections across 3 survey visits. So that's the total from 3 survey visits, the detections within these states. So the lion's share, Arizona, really is the winner of, of yellow-billed cuckoo distribution for our study. New Mexico had a significant number of cuckoos, 99. And again, that's excluding the Rio Grande population, which is probably the highest density of cuckoos in the state. California also had a significant number of yellow-billed cuckoos. And again, we did not conduct many surveys on the Rio Grande, on the lower Colorado River. So there's probably the largest concentration of cuckoos in California. Colorado had a fair number of cuckoos, as did Texas. Now, for Texas, we had to exclude sample units for a significant portion of the Rio Grande. A lot of our modeled habitat fell upon right on the border here, and multiple people advised us that that was unsafe to conduct surveys in those areas. So all of our sample units down in Texas were found down here in the Big Bend area. There were 5 that were in Big Bend National Park. There were 4 outside of them. So to zoom in just a little to our, well, let me just state real quick, only 1 cuckoo found, actually I think there's 3 cuckoos that were found at 1 location in Idaho, a single cuckoo found in Wyoming, 2 cuckoos found in Utah, and Nevada only saw 6 cuckoos. In California, the majority of the cuckoos that our project found were in the Kern River Valley. I think it was 48 detections in the Kern River Valley, which I'm not sure if you can see my mouse, but I'll pull up the laser pointer. The Kern River Valley is this star right here. So most of the cuckoos that were detected in California were detected right here. There were a significant number of surveys up in the Sacramento River. We found no cuckoos there. To zoom in just a little bit, again, New Mexico had a little bit over 50 sample units surveyed, and Arizona had approximately 110. And this is the distribution of those sample units within those 2 states. And here we saw where we had detections within those 2 states. Most of the detections in New Mexico were in these rivers around the Gila Mountains. So the Gila River in San Francisco, the Mimbres, I think that's still part of the Gila. So most of our detections were over here. Reclamation did throw us a bone, and they did let us survey a small section of the Rio Grande out by Socorro, New Mexico. We had a few sites with a strong number of detections. But as you move north in New Mexico, I think this represents 5 total cuckoo detections. And we had 1 cuckoo detection further north in northern New Mexico. This is north of Española, New Mexico. And we had 4 sites along the San Juan River in northern New Mexico where we had 1 territory. I think that's just 2 detections up there. We were particularly interested in what would happen in the boot heel of New Mexico, knowing that southern Arizona has Interesting habitat, and we presumed significant cuckoo abundance, which we saw. And we wondered if the same would happen in southern New Mexico, but the habitat just is not the same. The habitat in southern New Mexico and the Boot Heel does not get the monsoonal rains that southern Arizona has. It's very xeric. It's not part of the Sonoran Desert. This is very different habitat in southern New Mexico than was observed in southern Arizona. Nick Beauregard was our SSRS representative who oversaw a significant amount of the surveys here, along with Tracy McCarthy with Arizona Game and Fish. He's got a talk later on. He could probably give you a lot more information about the findings in Arizona. It's not a state that I'm as familiar with. We have not yet done occupancy assessments with this data, but looking at the raw data, looking at a quick selection analysis, we quickly see that approximately 40% of our sample units in New Mexico and about 52% of our sample units in Arizona were occupied by at least one cuckoo. Okay, our next steps. So these go back to our 3 primary objectives. So our first objective was to create a species distribution model. Utah DNR will be overseeing this. They're going to take our data from 2022, fold it in probably with the historic data they used to create the initial SDM, and we'll see what we get. For objective 2, we're going to do a 2022 occupancy assessment using the detection, non-detection data, incorporating detection probability, and hopefully a few covariates, and hopefully incorporating additional data such as from reclamation that we will increase our sample size maybe up to approximately 400 sample units. And lastly, California Fish and Wildlife Service will evaluate the ARU detection probability data, comparing it with our survey results in those areas. A big thanks to everyone involved in this project. Can't acknowledge how large this was and how many different partners pulled together to make this happen. This was a CSWIG project funded by Fish and Wildlife Service. We had state coordinators and people in all states within the DPS. And I'd like to give a special acknowledgement to Neil Clipperton with California Fish and Wildlife Service, Russ Norvell with Utah DNR, Aaron DeVuvier with New Mexico Game and Fish, and Edwin Juarez with Arizona Game and Fish. These folks have been involved in this project from the get-go. The first time we tried to apply for this grant was in 2016. And so these people have hung in this for a long time and have been a part of this every step of the way. All right, that is everything. If you have any questions, or if I have time, please let me know.
Hi, thank you so much, John. Some really exciting results. I'm so happy that that you guys have done this and that we have this picture here of where cuckoos are across the West. We don't have time for Q&A, so please put your questions in the chat.
There are some questions for John in the chat.
Okay, do you want to read one?
Sure.
Thanks, Jenna.
Did any of the occupied areas surprise you based on size of the polygon occupied? What was the smallest occupied polygon? Was it consistent with what is in the current protocol? You still here, John? Okay.
Yeah, I'm still here. We haven't quite looked at that fine a scale yet. So I haven't looked at polygons of habitat or patch size. Our sample units were all 1K by 1K, and that's the extent to what we looked at yet. So can't answer that. I just don't know. Good question though.
Is there another quick question for John? We try to address one of those, Jenna?
I see in the chat that cuckoos were found in the Sacramento Valley. That is fantastic. As part of our occupancy assessment, we're not going to use eBird data, but we are hoping to scoop up survey data collected in 2022 and at least show that on a map to at least create a visual picture of where cuckoos were found in 2022.
Great.
Laura, do you know what dates those cuckoos were found and if they hung around for the entire breeding season?
Mirlet, the cuckoo that was off of Little Dry Creek Farms, it was about mid-July. Unfortunately, that survey zone is a 2-person survey area, and I wasn't able to do it on my own. The area I was surveying for this survey method was just north of that, northwest about a mile from where that detection was. And it's a pretty dead-end habitat where it follows the drainage of Little Dry Creek that comes off of Butte Creek and basically goes into a duck club, and then all the habitat terminates around it because of the rice fields surrounding. So it probably bounced around on Little Dry Creek for a little while as it was leaving. The one on Calusa National Wildlife Refuge, I don't know when that one was reported. I only just heard about it last week from the refuge biologist. Jenna Zola was talking to my waterfowl biology class and mentioned it casually that she had several bird watchers that were letting themselves into an area after it had been reported that they should not have been commonly going into. And so that's how she had kind of discovered it, was by trying to figure out why so many bird watchers were sneaking in on that section of the refuge.
And we're going to move on to Jim Hatton's talk. I can see it. Good. Yeah, there
Can
it
you
is.
guys see this?
Yes, thank you, Jim.
Okay. So, I was asked if I would do a little comparison of the yellow-billed cuckoo and the flycatcher models that are out there, and I said I would. And it's interesting because, we haven't spent a lot of time comparing these two things. They were developed under separate efforts, but I just want to say the collaborators for the yellow-billed cuckoo were Matt Johnson, Jennifer Holmes, Patrick Shafroth, and then for the original flycatcher publication, Charles Pradzik was my co-author. Although Trey McCarthy, who's out there, and a lot of other people helped out, with the field work, and it was a tremendous effort to develop these models. So I just want to acknowledge that. And also U.S. Bureau of Reclamation of Fish and Wildlife and the Department of Agriculture, they've funded these efforts. Okay, so I just want to reiterate the Southwestern willow flycatcher model was published 18.9 years ago. Gulp, long time ago. It's going to be 20 here very shortly. And it's been used extensively by USDR and Fish and Wildlife Service. And it's being used more now, quite a bit more now than I think it ever has been. We did do range-wide modeling in 2016, which kind of expanded on the overall effort. So that worked out very well. We put out a report of that. And based upon that range-wide modeling effort, the U.S. Department of Agriculture, APHIS, they currently fund the using the flycatcher model annually. We update the website, the Swiffle Habitat Viewer, which I make available to everyone, and now it's available to you guys because I started sharing the cuckoo data on there. Now the yellow-billed cuckoo model was formally published in 2017, even though we had a couple reports out on it like 7 or 8 years earlier than that. But we did feel important to push it through and get it into a journal. So that was funded by USBR and Fish and Wildlife Service. The big difference between these models is that the yellow-billed cuckoo model has been used very lightly over the last 5 years. It's been used in Idaho, being used in the Willamette Valley this year in Oregon. It's been used on the Santa Clara in California as examples of its use, but it's pretty spotty, no, concerted effort like I just saw with the previous modeling effort. But anyway, just want to lay that out there. Am I still being heard okay on the audio?
Yes.
Okay. All right. I just wanted to point out there's a lot of similarities in the model in that they're both NDVI based, normalized difference vegetation index, which makes sense, right? Because riparian vegetation is green and lush. So they're based upon the normalized difference vegetation index, both of them. But they have differences. So the yellow-billed cuckoo has basically 120 meter radius core density area. And then it has a larger 480 meter radius patch, which is 72 acres. And that information from that larger patch is captured by the satellites. We look at the heterogeneity and the density of the vegetation and to feed that into a logistic regression model. So the take-home point here is that each 10% increase in the core area covered in dense vegetation we found related to a 15% increase in yellow-billed cuckoo. And then for each 1 standard deviation increase in the vegetation heterogeneity within that larger 72 acre area, there was a 68% increase in yellow-billed cuckoo. So that's good information there, I think, for restoration and enhancement activities. And when we did our classification accuracy, we were kind of in the 75% range. So is it it's because the cuckoo is a difficult bird to model. It really is. We also found that, that the cuckoo wasn't crazy about tamarisk. And that was a big difference in the flycatcher model. We have a 20 meter radius patch. So that's around 4.5 hectares. Okay, so it's a lot smaller than the cuckoo patch. That's the big difference. But we also found that for each 10% increase in that patch covered in dense vegetation, there was a 60% increase in flycatchers. And for each 1 standard deviation increase in that vegetation heterogeneity, 90% increase in flycatchers. So these models contain the information that can be used by managers to try and restore and enhance these sites. And then the big difference, of course, is that the flycatchers do utilize tamarisk for nesting. So that kind of separates the two out. The flycatcher model typically has 80 to 90% accuracy. So it's a little more accurate than the cuckoo model. So those are the main differences, but they're both driven by Landsat. I'm running them both right now on Landsat 9 successfully. I was running them on Landsat 8 last year, and Landsat 5, in which these models were developed, they're not even on Landsat 5 no longer exists. So I actually I have a routine that harmonizes the Landsat 9 NDVI data so that it looks back in time and creates similar NDVI value as the Landsat 5 produced. That's a real important thing. So I also want to say that I think that these models are underutilized. I want to show you an example. Let's see if this works, where you can actually create video movies and see how the things change over time. So here's an example of Landsat imagery that is from 85 to 2005. And that's the kind of the Winkleman area of the mine there that you see on the lower right. So it's where the Gila River and San Pedro River come in. And then on the right is the exact same area, exactly, but it's the output that the model produces when this satellite model runs. So I'm going to see if I can run both of these at about the same time. Let's just see. You can watch as you see the big burns in the riparian areas and how they're reflected in the actual habitat models. You're going to see a huge flood. That's the 93 flood there. You see how it's very well reflected in the imagery and the mapping. So you can actually see how things change over time. The wet years, you see that like bigger expansion of the lateral margins of predicted habitat, although the model's saying that's not habitat, but it's saying that it considered it. The yellow areas the model considered, but said, no, you're not habitat. So this can be done for any reach, you know, where you can look at the burns, the floods, effects of drought, and insects. So, and lastly, I've placed the yellow-billed cuckoo predictive model now on the public flycatcher habitat viewer. I'm doing that as a freebie and a favor to this group. You know, I asked the Department of Agriculture if that was okay. PI doesn't have a problem with it. So now, and then I went ahead and I just joined now the flycatcher and the yellow-billed cuckoo predictive models together. You know, I'm not merging probabilities. Remember, I'm just taking the habitat maps and joining them together and producing a map now that are either flycatcher habitat predicted or yellow-billed cuckoo predicted habitat, or you're both. You overlay each other. So, and found so far in doing this is that there's a 40 to 45% overlap. I've done it on several years now. There's a 40 to 45% overlap in flycatcher and cuckoo habitat. Okay, so, and I just wanted to reiterate that it's possible to expand this, you know, range wide if people want. I know there's another modeling effort going on, but just letting you know that it can be expanded. Right now, it's the range of the flycatcher, just because I'm piggybacking on the efforts of that modeling. Reach-specific analysis, I think you should visualize and quantify them using the videography method that I showed you. I do think combining the models increases the net value of what we're doing out there, because now you can actually even survey areas where you can, where both species habitats are predicted suitable. Right now, there's no charge for running this model. For the range of the flycatcher, I'll continue to run it and just add it into the flycatcher public viewer for you. If you want to expand beyond that, then, you know, people have called me and I've done little favors here and there and ran it in various locations. But if there's any concerted effort, then we would have to have a further conversation. And then I thought what I would do here is actually go into the habitat viewer. I know, not sure where my time's at here, but let me go ahead real quick here. This thing is actually in my way. Hang on a second. Okay, let me bring up the public viewer. See how this works in real time. I ran it just a few minutes ago and it ran really fast. You know, I'm on a satellite connection where I'm at, but let's see how it comes up. So, takes a minute to load. I wanted to do it in real time. I wanted you guys to see that, because that's available to all of you. So the link it's up on my end now. Let me know when you can see it. See if this works. I see a green bar around it now.
Yeah, now we see it. I see it. It looks good.
Okay, good. All right, so, and this is, remember, this is the public habitat viewer for the Southwestern willow flycatcher, but I provide this layer now that any of you have access to. And it's what I've done also, I've done some enhancements to the database. So I'm going to just select a patch here. You can see when I select that patch, it gives me information. So right now that COM ID, that is the ID of a river reach that's in the NHD Plus database, the official database for the whole United States for that reach. So far, I'm not aware of anyone really utilizing that, but I put it there in case people want to be able to link to the NHD database. It gives the name of the river, and then that Q0001E, that comes from the NHD database. What that is, it's just an estimated annual stream flow using the closest gauge data to make it as accurate as possible. That's what the 1E means. And then below that, V0001E, that's the estimated stream velocity for, and I think it's feet per second for that reach as well. And then it gives the patch, and heck, this one, that's a discontiguous cell there, 0.09. That means it's a 30-meter cell. But you know, if you just click on other patches, you'll get different results. So I've made that available to you now for anywhere this model is run. And one thing I like about it is the search feature. So let's go to Patagonia, Arizona. Let's just go there for a second, because it's really a nice feature of ArcGIS Online. It's really quite fast and snappy. So now we are on Sonoyta Creek, and if I just select the patch there, so this is saying that this is 87.3 hectares. And if you look below, it says describe. That's the attribute. If it says Swiffle, it's only Swiffle predicted. It says yellow-billed cuckoo, it's only predicted cuckoo habitat. But if it says both, like this one, it's saying that both these models concur that it's cuckoo and Swiffle habitat. So this is a gigantic patch right here, 87 hectares, where it's saying it's both. So, and then the last thing, I know, I guess I'm about out of time, but let me just show you real quickly here. There is the screening tool. That little screening tool is kind of handy, where if you click that, and then you go up to your draw feature, you can click the shape. I'm going to click a square for a second. I'm going to draw a square around me here. And then if you select report, you'll see all these layers come up and they got these red exclamations. That just means I'm not visualizing them in the map. I can turn those layers on. But if you scroll down, you'll see down below that the layer that combines both the flycatcher and the cuckoo, that's the layer it's showing. It's saying there's 44 distinct patches in this square. So any cell that's separated becomes a patch, unless you change that. But if you open that up, it'll show you the patches. I'm having a little bit of a bandwidth thing here. So, but on your end, it will show you all those patches, the size of them, etc. And then up above, you see that down arrow there? If you click the down arrow, it allows you to export this data. And you can export it as a shell. And if you click download, it will clip that and download that to your computer for you. So anyway, these are the main features. I know I'm out of time. You know, you can change your background layers as a base map gallery, that type of thing. So that's it for me. Does anyone have any questions?
Sure. Wait 1 second. Thank you so much, Jim. I really appreciate it. We are out of time for questions. We're supposed to break for lunch right now, and then have lunch for about a half an hour, and then come back at 12:30 to have our afternoon session on characteristics of breeding habitat. But I just want to let everyone know that I'm not going to close out this meeting during lunch. So folks can stay on, ask questions, talk amongst yourselves, have a discussion. The lines are open. So if you need to go, then we understand and you can go have lunch. But if you want to stay and talk, the lines are open. So thank you so much to all the presenters this morning. I really appreciate it. And thanks to you, Jim, for hanging in there and getting this going, getting your presentation up. I appreciate it.
Yes.
All right. Are there any questions to Jim? Tracy, I see your hand up.
Yeah, Jim, I was wondering what it would take to expand the cuckoo model out into the Zara riparian habitat in Southeast Arizona that's not these typical perennial drainages.
It would just take a shape file, you know, of the reaches that you're interested in. We would have to, you know, apply some sort of buffer to it. And then it's, you know, it's good to go. We could run the model on that. So
Okay.
if you wanted to do that and you had a shape file and you wanted to send it to me, you'd do me a favor if you buffered those creeks in advance for me.
Right.
Maybe try to include areas that you want included and exclude with a polygon layer, you know, using ArcGIS or something, the areas you don't want, whatever, and send it to me, and I would certainly explore that with you.
Okay, I also think it'd be interesting to use the data that we just collected in those perennial drainages for the Rangewide model and kind of ground truth, the Cuckoo model as well. So we might talk about that a little more later as well.
That would be great. I mean, we haven't had a lot of feedback. Like I say, it's been used 5 or 6 times, but not extensively. And our really the feedback for detections has been pretty sparse, because the areas that it's ironically the areas it's been applied to haven't been heavily occupied by cuckoos, like the Santa Clara and Idaho, and now up on the Willamette in Oregon. And I'm not even sure what was found with that effort. I did run the model in the Willamette. So, but those are areas with very few cuckoos, so we've received virtually no feedback. So anyway, I'll work with you guys as best I can.
The one thing, Jim, I
if-
just wanted to emphasize that we are applying this model, both the yellow-billed cuckoo and the Gila, and the yellow-billed cuckoo and the Swiffle model to the Cliff Gila, and we're working with the Nature Conservancy on that, and that's
In
an
New Mexico.
ongoing project that we're
Yeah.
doing right now.
New Mexico.
That's true. That will be some good verification data there. So, you know, we're getting out there. You know, we don't have the effort or the resources as the previous group. But having said that, it would be great if there's a little more collaboration between the two groups. I mean, our model's out there. You know, it's available for people to use, and it's free right now for the range of the flycatcher. But if we can overlay the new model that's being developed with ours, to me, it's just value added. I mean, you look at the really good sciences, you know, like you look at the National Weather Service every night, I'm so inspired. They predict with multiple models, you know, and uncertainty, they actually can quantify by the agreement and disagreement among the models. So I just see it as a win-win. So anyway, our model's out there and we'll collaborate with people.
Jim?
Definitely.
Sorry, there's another question from Bruce Orr.
Yes.
Bruce?
Sure. Hi, Jim. I just thought since we've got a little time at
Yeah.
lunch, maybe you could briefly mention your past applications of LiDAR data to add canopy height to your models, and sort of maybe just a quick pro and con summary of that approach.
Yeah. So anyway, we did on the Gila around the Safford area. I don't know, it's probably getting close to 10 years, 7, 8 years. I lose track of time, but there was LiDAR data flowing for the Gila, and we had elevation heights. And I did enter that into the flycatcher model and explored how vegetation height affects the model. And it did improve classification. Our ROC curves did improve. It was statistically an improvement. So that was all good. The problem with LiDAR data is its resolution. It's so darn high resolution. So I had to aggregate from sub 1 foot level to, I aggregated it to 4 meter pixels just so I could work with it. And then those 4 meter pixels, you can fit like 25 of those into a 30 meter cell. You know, so I then had to resample the satellite model in order to get the two at the same resolution. And then once I ran it, yeah, there was an improvement. The problem is it also makes, though, the model becomes noisier. So you're more likely to be able to find good habitat, but the patterns and the patches begin to break up and become more speckled and noisy. So I'm sure there's a lot of approaches to dealing with that, filtering, that type of stuff. But it really is an area that needs more work. Just absolutely more work. But am I a believer that veg height increases the predictive ability of models and improves quality of models? The answer is yes. Just getting it to work and work well.
Thanks, Jim.
We could explore it on the Santa Clara. Just let me know.
Are there any more questions for Jim?
Okay, I'll let it.
Is there anything in the chat, Jenna, if you're still here? You haven't left for lunch?
No.
Let's see. Oh, I see. Could you put the link for the model in chat? I'll see if I can manage to do that here after I hang up here. It'll be the Swiffle habitat viewer. Okay, I've got the link in there. I'm going to send it to everyone. Did it send?
And
Maybe I
it's,
got to hit return. I don't know.
oh, there it is.
That is.
Yeah, maybe it just had to go to everyone.
I'll just take you to the public viewer, habitat viewer. Now, you know, I did create a private Cuckoo site, and I had about 15 people signed up for it. But I realized that we had an opportunity to piggyback on the public habitat viewer for the flycatcher. And once I merged the two models together, it made sense to do that. So I haven't done anything else on the private viewer. But we could make that private viewer public if anyone wanted it to be more of a nexus for any type of spatially explicit modeling, maybe the type of stuff with Chasey McCarthy and some of these various locations. I don't want to put that stuff on the Swiffle public viewer. So I might ask USGS for permission to make the site public. Or we can keep it private. Maybe people don't want their data out there. So anyway, we can talk about it.
Tracy, did you have another question?
Yeah, Jim, I was wondering how hard it would be to put a base map layer of the satellite imagery on there.
Okay, so, and when you say on there,
well,
do you
on
mean
the
on the Swiffle habitat viewer?
Yeah. So you have the topography map on there now as the base layer. How hard would it be to add an aerial satellite imagery?
So there is a button there. My bandwidth's a little low right now, but you'll see that little button that it's the base map gallery button, the four squares. Yeah. And that, if you click that, you can add any of those layers, including the latest Esri. You know, Esri, they usually update their imagery every year.
Oh, I just, when I open that, I get a blank box.
Yeah, that is interesting. And I'm not sure, I mean, usually it works perfectly. I'm not sure if that's coming from because my bandwidth is low right now, although it's a cloud-based program. So I don't know. I'll have to investigate that. Normally it works perfectly.
I've tried it before because I use your model. I do.
Yeah.
And I just never, every time I click on it, I don't see any other base map layers.
Okay, I will go in and investigate that today. Like I say, I've used it many times, but I noticed last time I used it, it was blank. So
cool.
I might have to contact Esri. It's a cloud-based resource, you know. So, but I'll get it working.
Great, thanks, Jim. I'd appreciate that.
Thanks, Jim, for staying on. And we have about 15 more minutes of the lunch break. But we'll have a little bit of time there when we set up the afternoon session and hand it over to Kristen with Peak, so people have a little bit of wiggle room there. And so thanks everyone. And if you want to stay on and talk, you can. I'm going to step away from the screen for a little bit and I'll return back in 15 minutes.
Hi, everybody. My name is Kristen O'Hara, and I am the director of interpretation and programs at Pajarito Environmental Education Center, or PEAK, as we like to call it, where I'm from. I will be the moderator for this afternoon portion of your working group meeting. Hi, nice to see everyone. PEAK, if you're not aware, is a nonprofit that operates the Los Alamos Nature Center. It's a lovely little nature center located in Los Alamos, New Mexico. Please come and visit. For more information about my organization or to make a donation even, please visit peecnature.org. That's peecnature.org. All right. So just a gentle reminder, which I'm sure everyone got this morning, everyone who attended, that is, please be kind and respectful to your fellow attendees throughout the duration of this meeting. We will, of course, begin this afternoon session with lightning round presentations. Those presentations are about 5 minutes each. I will be keeping time, so whoever is a presenter out there, please just know I will probably interrupt you right at 5 minutes and let you know. If you are in the audience and have any questions, please write your questions. If they're like burning questions that you absolutely need to get out there, please put them in the chat. Or if you can save them, please save them for the discussion portion of this afternoon meeting, which will start at 3:10. This meeting, of course, is being recorded, and we will post it on Peak's YouTube account. We will let everyone know who has registered for this meeting where that and when that is up. Okay, so last thing, if you are a presenter that will be giving a talk, please make sure you put your first and last name as your Zoom name. So go ahead and rename yourself if you, for instance, are labeled as a phone number or something like that. All right, so it looks like our next introductions are with Hira at 12:40. Hira, I don't know if you want to start that now or if you just want to give it a couple more minutes.
Kristen, I think I'm going to give it a couple more minutes because we actually broke for lunch a little late. And so I want to give people some time to come back, and then I'll give the introduction to the afternoon talks, and then we can head into the lightning talks on time.
Sounds good. We'll start at 12:40 then.
Hello, everyone. Welcome back from lunch. I'm just going to quickly give an introduction to the afternoon presentations. So as you know, As you know, the meeting topic is range-wide and regional characteristics of yellow-billed cuckoo breeding habitat, what we know and what we have yet to learn. We decided to have a meeting on this today because we feel strongly that accurately identifying the abiotic and biotic features of cuckoo breeding habitat is vital to cuckoo recovery efforts. There are resources out there that you can go to and look for information about what is yellow-billed cuckoo breeding habitat. For example, for sort of range-wide habitat, you can consult the proposed and final rule on critical habitat. In that proposed rule, they mentioned that there's 3 primary constituent elements of yellow-billed cuckoo breeding habitat. The first one is riparian woodlands, specifically riparian woodlands with mixed willow cottonwood vegetation, mesquite thorn forest vegetation, or a combination of these that contain habitat for nesting and foraging in contiguous or nearly contiguous patches that are greater than 325 feet in width and 200 acres or more in extent. They also mention that these areas are generally cooler and more humid and have above average canopy closure. Another primary constituent that they mention is adequate prey base. The availability of prey such as cicadas, caterpillars, katydids, etc., and even tree frogs are important for cuckoos and an important predictor of occupied cuckoo habitat. The third and final primary constituent element that they mention in the proposed rule is dynamic riverine processes, specifically river systems that are dynamic and provide hydrologic processes that encourage sediment movement and deposits that allow sealing germination and promote plant growth, maintenance, health, and vigor, which allows habitat to regenerate at regular intervals, leading to riparian vegetation with variously aged patches from young to old. As we saw in John's talk this morning, though, cuckoos breed in dynamic ecosystems across a wide geographical and elevational range. And so, correspondingly, their habitat varies across their range. We really want to understand this variability. And so we have invited folks from 9 states, 9 US states, and from Mexico to come today and give you 5-minute lightning talks. We've asked each presenter to give information on the abiotic and biotic characteristics of cuckoo breeding habitat within their region at 3 scales, at the landscape scale, the patch scale, and the nest site scale. And that's only if they have that information. Some of this information is not available for particular locations. We also want to understand, how do we measure that variability? And we had some nice discussion about that over lunch and also with Jim Hatton's talk. But later on today, Bruce Orr will be giving a talk about using LiDAR to assess the vegetative structure of potential cuckoo breeding habitat. After all of the presentations, those lightning talks are completed and the main bulk of the presentations are done, we're going to have a discussion on range-wide and regional characteristics of cuckoo breeding habitat to really get into discussing some of that variability. Specifically, we hope to discuss what patterns and habitat characteristics we see across all regions, how we should delineate ecoregions of similar breeding habitat for management recommendations. For example, are breeding habitats within New Mexico more similar to West Texas, or more similar to southern parts or western parts of Arizona? How do we delineate these ecoregions of similarity? And then we also want to talk about, when we're trying to make management recommendations, which habitat components are most useful at the range-wide level and then within these ecoregions. We think that the results of today's meeting will have wide-ranging applications, but specifically, we think that it will help to inform the preparation of both range-wide and regional guidance documents and recommendations for cuckoo habitat protection, creation, and restoration. And more specifically, we hope that this information will inform efforts by Muriel Halterman and her colleagues to revise the cuckoo survey vegetation data sheets, specifically to help those data sheets be more effective at capturing the most significant habitat characteristics and also increase the usefulness of the data collected. We don't want folks to go out there and do all the hard work of collecting vegetation data and for those data to just sit on a shelf somewhere and never be used. We want those data to be useful. And so identifying some of these important characteristics of cuckoo breeding habitat should inform what data we should be collecting in the vegetation data sheet. Also, the results of our discussion today will hopefully inform efforts by Jay Carlisle and others to develop guidelines for breeding habitat protection, management, and restoration. Right now, they've drafted a range-wide handout, and they're still trying to fill some information gaps, like how contiguous should riparian habitats be to support movement of foraging cuckoos. Hopefully today we can help identify those regions of similar habitat characteristics, those geographic regions, and then identify partners to develop restoration guidelines for those regions. So Jay et al. Will be working kind of on a range-wide guidelines, and then we hope to get so folks identified who could do some regional guidelines. And Jay's going to talk about that right after the lightning talks. And so with that, I'm ready to start the lightning talks.
All right, so it looks like our first presenter is going to be Edwin, and he will be, his presentation is about statewide habitats in California. Edwin, take it away.
Hello, everybody. Can you guys hear me and see my screen?
Yeah, yes and yes.
Okay. Thanks for the introduction. So yeah, I want to be talking about California. And California is such a large place to summarize in just 5 minutes, but I'm trying to do my best. These are some of the most important current and historical populations of yellow-billed cuckoo in California. As John mentions, the Southwestern River and Lower Colorado River are two of the biggest one. And as you will see, my presentation is mostly focused on those because there's more data on these populations. In the landscape scale, we have that there's a lot of variability in how these lands are managed. There are, for example, in the Lower Colorado River, you have water management to enhance wildlife. In the Owens River, you have water that is just carried through the landscape to fulfill the needs of LA area. And you have in general this variability and influence of dams and other water management that alters the regime of the hydrology. In terms of vegetation, we have mixed riparian forest, mostly dominated by willow species, mostly Gooding's willow, but also red willow, and also cottonwood, mostly Fremont cottonwood and black cottonwood. But cuckoos have been known to occupy, at least historically, some walnut orchards. In the surroundings of these riparian forests, we have typically shrublands, pasture, agriculture, and even some urban, like small cities, mostly in the Sacramento River. The riparian forest is typically wider than 100 meters. But again, this is highly variable because we have places where this narrows. We have also places where the agriculture and the pasture get embedded inside the forest. So it just kind of like breaks the connectivity of these riparian forests. And the hydrology in some places is highly variable, like in the Kern River Preserve, in the Kern River Valley, we have this the hydrology and the water flows depends entirely on snowpack. So it's highly variable among years. And I'm going to show you some photos of these areas. We have here some photos of the South Fork River, the Sacramento River, and the Owens River. And you can tell how these are these strips, very wide strips of vegetation that goes from, again, from 100 meters, can get narrower, but can go up to 500, 800 meters in some places. And again, these are along canyons where you can find these alluvial plains that are under the influence of the rivers that go through. Here we have some photos of the Santa Clara River, and again, it's the South Fork River. That is very special case because we have the Lake Isabella that goes up and down and floods some places of the forest that get under the influence of these floodings, and then it drives the replacements and the successional stages in the forest. When the water is high, it can kill some trees, but then when the water is low, it promotes regeneration of the forest. In terms of patch, it's not required for cuckoos here to be close to water or to be in places where the soil is saturated. They can be away from the water. And in general, they have a preference for young stands, but they can also use mature and a wide range of successional stages of forest. Again, they use willows, cottonwoods, mesquites in some areas in the lower Colorado River. In the understory, they typically, in the sites that they choose, there are stinging needle, mule fat, and other species. And in general, we have a dominance of native species with a low cover of salt cedar. The vertical structure is very dense forest, typically over 50% of the canopy cover, and the height of the trees is highly variable. They can go from 4 meters to up to 25 meters and even beyond that. And again, the separation of these trees can be also very variable, can be very dense stands or can be sparse trees. But as you'll see in the nesting situation is a little more complicated than that. They choose more dense areas. Here we have some other examples of how this vegetation looks like in the South Fork River, Santa Clara River, and Sacramento. This is the South Fork River, and we have this regeneration that came after the Lake Isabella levels went down, and this was prime cuckoo habitat in the 2019 and 2021 breeding season for cuckoos. So we have a lot of pairs in this area. And finally, for the nesting site characteristics, it's highly variable. They don't need to be close to water. They don't need to be in places where they is soil saturation. They have been known to be nesting in willows, cottonwoods, in mesquite, and also in salt cedar in lower Colorado River, and in walnut, apparently in the close to the Sacramento River. they typically nest in live trees, and the nesting location is between 4 and 26 meter, which is highly variable. But they tend to choose very dense areas, as you can see here. So I think this is John Stanek looking for checking a nest in the South Fork River. All these photos are from the South Fork River, but you can tell how dense are these places where the nests are. This nest here is very concealed. You can barely see this nest from the ground. You cannot see where the nest is here, but it's somewhere in this tree. And here are more examples of nests located in willows. You can tell, like, it's very challenging to find them because they are very concealed. They're very dense places within the trees. And that's all I got. And thank you for the opportunity for talk today.
Awesome. Thank you so much, Edwin, for that. I believe our next presenter is Merlette Halterman. And sorry, Hira?
Oh, we have just a couple of minutes before the next talk is supposed to start. 3 minutes till the next talk is supposed to start. So I just wanted to see if anyone had a question for
Okay.
Edwin.
Yeah, definitely. Edwin.
Anybody have a question? A quick question for Edwin? Is there anything in the chat? Bruce, you have a question?
Yeah, I was just curious about what Edwin might be able to tell us about the population sizes coming from this last year's surveys, especially the Kern River. I'm curious as to how many birds are now breeding there.
Yeah, I want to say that they need to be in a patch that is at least 100 meters wide. They tend to move a lot when they are feeding their babies. So I will imagine that it's also context dependent. If you have several patches that are probably not optimal in size, but they're next to each other, you can probably have cuckoos in that area. So I think it's difficult to tell a pixel size to survey or a patch size to survey if, like, isolated from context of where that patch is. So I don't know, probably John has a better answer to that because he's been analyzing this data for longer than yeah.
That's fine. We can follow up later on that, but I definitely would like to talk to you guys more.
Sure.
Awesome. And with that, we're going to roll to the next presenter. Thank you so much, Edwin. And our next presenter is Merlette Halterman with habitats along the Owens River in California.
So, are you seeing the show now?
Yes, we can see the title slide.
Perfect. Okay. So, Great Basin Bird Observatory did surveys on the Owens River for 2 seasons, and there was some difference in the areas that I was surveying, but basically between Bishop, or mostly near Big Pine. So Edwin and Southern Sierra, they gave a good overview. I'll just give a little more detail of where I surveyed. So again, it's east of the Sierras, and this is important. This area has a lot of riparian, and there has been populations here, although they've only been intermittently surveyed and studied, but there have been cuckoos here for a long time. So again, you can see this is all pretty much, all that green is cottonwood willow. And then you have the Owens River with fairly narrow band habitat with a little bit more down at the southern end, just above Tinnimaha Reservoir. So we did 4 surveys each year and 1 detection last year and 5 detections this year. Plus there were 3 additional detections. One of these was probably a breeding pair. And I wanted to talk about this because this shows the variability and with the geolocators that these birds are moving around a lot. And there are cicadas in the area, and we definitely heard more cicadas this year than last year. So although there may not have been many birds breeding where I was surveying, there were birds. There were certainly birds, in the area. I'm trying something else because I can't see the chat. Anyway, and let's see, let me go next. This is the Owens River. Each of these colors represent the survey area, survey route, and tried to cover a lot of habitat. Owens also worked on Baker Creek and Hogback Creek. So again, variety of ownership of the properties of the land, cottonwood willow. Again, fairly narrow. With the zoomed in shot, you can see that, sorry, I don't have a scale here, but this is sometimes, 50 to 100 meters wide. And you can see all these oxbows. Historically, there's been a lot more riparian along the river, but with the watering of the Owens, also the river's deeply incised, so the vegetation is not what it was 100 years ago. It's another picture. This is a wider area, and this is the area where I had 2 detections, one in June, one in August, indicating that there may have been bird breeding in this area. So again, you can see a lot of sagebrush here, right on the edge of the cottonwood willow. And again, rabbit brush and sagebrush. And a permanent flow. There is some cutting here, but these banks are often 4 to 8 feet high. So the river, as I said, overall is pretty up. And of course, it's between the populations of the Colorado River and the Sacramento, a bit north of the Kern. So it may be along a migratory path with the birds. And so they check it out some years and aren't there others? As I said, perennial river, you can just barely see the water. There are storms if that happens and the soil's saturated. Otherwise, it's very dry out there. And all this is just basically salt flats and scrub vegetation next to the river. And not a lot of regeneration, a little bit, but not a lot. And so you mentioned cottonwood willow. There is a lot of emergent vegetation. There's a lot of sandbar willow, and I can say that vegetation, and it is mostly multi-structured. And I believe that's it. As I say, I have a fairly short presentation here, so I'm going to switch to. Well, actually, I can hear your questions. You may not be able to ask me.
I'm so sorry that you're feeling atrocious. I hope you feel better. We actually are right at the timing for the next presentation. So perhaps we'll hold questions for you in the discussion round, or if we have time in a little bit.
I'm not sure, Russ, are you giving the talk for Utah?
Yes, I am.
Okay.
Perfect. Russ, go ahead and let me find you, and I will.
Yeah, I can raise my hand if that helps.
I got it. You should be able to co-host yourself now.
Okay. Well, I'm actually going to make this much easier.
Perfect.
I am just going to talk and not have a presentation exactly. So this is a lightning talk on the condition of riparian habitats in Utah. As you know, I think you heard in John Stanek's talk that we have had very few observations this year, and that's actually fairly typical, I would say, in some ways, of Utah. So it's hard for us to, I think, speak to any really consistent habitat use in the state of riparian. We have not seen that in many, many years. I don't even know that we have a breeding record in the last maybe decade or so. Most Utah riparian habitats are naturally narrow, especially in the southern portions of the state where these are red rock canyons. They're naturally narrowed, naturally disjunct frequently too. And unfortunately, these are highly regulated systems for the most part. We don't have a lot of opportunity, even when there is a flooding event, to have a lot of regeneration. So these tend to be small patches of native vegetation in areas where the systems are more dynamic. However, below dams, and there's a lot of water management in Utah. Those systems tend to be, over time, dominated by non-native Russian olive, tamarisk vegetation, which certainly does reduce vegetative structural complexity in these areas. That's kind of the local patch scale. We have also, of course, pervasive, I think, grazing pressure, which has helped to enforce the lack of long-term regeneration for native, especially for native species, which just plain taste better, I think. We've also, stepping out a bit, we've also worked with the researchers to use some remote sensing tools and modeling efforts to assess overall riparian habitat condition. This was a work that was completed some years ago and it's kind of laying fallow as a means of directing habitat restoration work. The indication was that a lot of riparian habitats, what we call low elevation riparian habitats, say below 6,500 feet, are in relatively poor condition and have been increasingly so. With the increased drought severity in the last several years, this has really come to a crucial point. So our riparian habitat conditions tend to be poor. As well as being naturally limited, they are also poor quality when we get there because of invasive species and just lack of water, really. That's kind of our fundamental there. We have not had any breeding activity outside riparian systems that we've ever noted, but we do have wide observations during migration dispersal periods up into the mountain shrubs that have never been surveyed for breeding efforts, so I'm not going to speak about those as habitats potentially. I would say that we lack any real data on prey-based information. That's one element that has escaped notice, I think, of our state in particular anyway. Other states maybe as well in a similar situation, but that's not something that we can really speak to. I think that would be a fascinating use of the ARUs and other kinds of tools to help assess some of the value of these areas for large-bodied insect prey potentially. So it's a fairly, I'm not saying this is an awesome system, but Utah is probably not going to be, and maybe never was, cuckoo, the heart and soul of cuckoo habitat. But when our systems function appropriately, I think, and provide that ecosystem function we do and can support a native cottonwood willow, typically Fremont cottonwood and mostly coyote willows and sandbar willows are our primary species here. These are the species that dominate in the areas where we do have our most consistent cuckoo use. Although, like I said, we have not been able to document actual breeding in the state for some number of years at this point. That might be as far as we want to go with it here today. I know it's not the most particularly rosy perspective. I will say that we have a state-run collaboration amongst state and federal agencies that look to do habitat restoration. And riparian restoration is one of the main targets of that group. However, accomplishing habitat restoration in our systems can be very expensive and not always successful. So we have had maybe less effort put there as we might like to see. So knowing where to begin would be a great opportunity. I'll take it. I'll stop there.
Awesome. Thank you, Russ. We are at the time for the next presentation, so we'll hold off on questions. If anyone has them, if they're burning, you really need to get them out, please put them in for Russ in the chat. Thank you so much. So our next presenter up is Elizabeth Eamon. Elizabeth, you should be co-hosted now. You should be able to share slides if you would like to. And Elizabeth will be talking about statewide habitats in Nevada.
All right, sounds good. I'm going to share my screen here. All right. So I'm giving you a status update for yellow-billed cuckoos in Nevada as of 2012. And you might notice that this is a little dated, but that was the last time we have actually done anything for yellow-billed cuckoos comprehensively in the state. I don't know why I can't. Okay, here we go. Okay, the methods were very straightforward. And by the way, Muralid was mostly doing all of this, so she should really be giving this presentation. But anyway, we looked at all potentially suitable sites in Nevada, which were kind of well known for people who live here and do statewide work. So we knew kind of where the places were, but then we also looked at all the historic records that were collected in the state that we could find at the time. This was kind of when eBird was just getting started in the state. And then all of the historic locations were resurveyed for yellow-billed cuckoos using the standard protocol. But then we also try to assess all the sites that were visited as for habitat suitability, just so we have some basic data on where places are that are potentially suitable or could be suitable in the future. And this is the map of all the locations that mostly Muralid visited, and some of which had cuckoos, and most of them did not. Then we had a data sheet to basically characterize in very kind of rank choice terms or rank order terms of how suitable, or of different types of environmental factors that we wanted to record, just because any of these places, we didn't know what the history or what the future of those places would be, if there was going to be any restoration in the future, that we would have some basic data on what these places looked like in 2012 when we did the surveys, and then also have some, maybe recommendations down the road of what to do with them. So these were the historic data, and I'm not going to bore you with the details, but you can see in the Great Basin portion of Nevada, there were historic data, not all of which were breeding records, but there was hardly anything at the time. So it was really, truly time for somebody to go back and look at these places. Now in southern Nevada, in the Mojave Desert, it looks much better from a historic point of view. There were a lot of data, and you can see on the Muddy River particularly that there were at times relatively good numbers of yellow-billed cuckoos. And the Virgin River is the other kind of stronghold in southern Nevada, or in, let's say, in Nevada. So, and these are the places where we found birds. Basically, this is the same map, but you see the red circles. The main one in the Great Basin is the Lahontan Reservoir Delta, which is kind of the outflow of the Carson River into a reservoir, and which has a very broad riparian corridor. And then the other place was Ash Meadows. And then there are two circles that you see down here in the southwest and the southeast. These are empty because we did not survey them, but our partner agencies did survey those, and those actually tend to be historically the strongholds of the bird. And they were again found there, not in the numbers that were historically recorded. All the other places had nothing. And that was it. I know that there are a couple of other folks from Arizona, from Nevada, I should say, on this call. And if I misrepresented anything, or if Muralid has anything to add, if there's time, she's welcome to correct me there. And as you can see, those data are 10 years old. So, since then, the historic sites have occasionally been resurveyed, but statewide assessment has not been done since then, as far as we know.
Got it. Thank you, Elizabeth. We have just a few minutes for questions. If anyone has any questions.
Elizabeth, can you describe a little bit the habitat in the stronghold area that you pointed to on your last slide? Is that
Yes,
typical to what we, okay.
Yeah, this is a good question. I probably have some photos of those places, but it's mostly cottonwood and then Goodings, willow, and sandbar willow, I think, are the main dominant trees, I should say. And some of them might have ash, like an ash meadows, for instance. But that's it's the kind of basic Mojave Desert riparian, but usually younger and more intact, if that's a good way of describing them, than what you see on average. So, and Muriel could probably add to that if this is not sufficient as an explanation.
She's put a lot of information in the chat right now.
Yes.
Oh, okay. Oh, is Muriel busy on the chat? Okay.
Yeah, she is. She's losing her voice. She's a bit of a cold, so.
Oh, okay.
It's not her forte today.
Okay, yeah. Then yes, the better answers are probably in the chat right here than what I had, because I've seen those places, but I didn't do the surveys at the time. And yes, actually, the Las Vegas Wash, if Debbie is not on the call, there were sightings. The current situation is probably very similar to what I just showed you in those maps. But there were sightings here and there on the Walker River, on the Carson River, and on the Truckee River. Well, not on the Truckee River as far as I know, but there were occasional sightings. But what I try to focus on is the places where we either suspect strongly that there have been nesting there for a long time, or are still nesting there, or where there's a good possibility that if even a little bit of restoration was done, they would be nesting there. So it's a pretty sparse situation across the state.
Can you hear me? This is Debbie Van Dormelen.
Oh, there's Debbie.
Hi,
Elizabeth.
Elizabeth, and I'll see you on the Nevada Partners in Flight call in just about 40 minutes.
Yeah, and I apologize for being late, by the way. I got the hours wrong. Anyway, go ahead, Debbie.
Sorry you're not feeling well. So yeah, let me chime in because with Southern Nevada Water Authority, which is who I work for, and there are a few of us on the call, we own the Warm Springs Natural Area outright. And that's one of the parts of the strongholds for the habitat in the Muddy River here in Southern Nevada. And then with the Las Vegas Wash, we are the implementation team for the stakeholder project that's performed the stabilization and enhancement program out there. We do pretty consistently have detections, including those that rate to probable breeding territories on both properties, and really just about every year. This past season was the first time in several years that we didn't have any detections at the Warm Springs property, but we documented a pair just a year or two ago, and pretty consistently have a probable breeding territory out there. But again, I will say it's one probable breeding territory. And then at the Wash, it's this past year, we had a couple different detections, seemed to be migrants, but we've had probable and possible breeding territories out there too. But just to put it in context, as Elizabeth showed, we aren't a stronghold for cuckoo, and perhaps we never were. Like that person in Utah mentioned, we just have so few detections that the few that we have at Warm Springs Natural Area on the Muddy River and the few that we have on the Las Vegas Wash, which flows into Lake Mead, are significant for our state. I don't know that they're significant for the species as a whole, but in terms of range and in terms for our state, we are significant sites. And it is Cottonwood, Willow, Warm Springs Natural Area has a substantial ash component. That's our site on the Muddy River. We don't really have a lot of ash on the Wash. It's very negligible, but both sites are really helped by the presence of mesquite, that adjacent mesquite.
Awesome.
Yeah, good point. I forgot about the mesquite. Yeah.
So we're actually running into our next presentation. So if we'd like to continue this conversation, please do so in the chat. It is very interesting. Thank you so much for all the extra information. So our next presenter is Jay Carlisle. and Jay will be giving an update talk about the statewide habitats in Idaho and Wyoming. Jay, you should be co-hosted and you should be able to share slides if you would like to.
Okay, is that showing up okay? And can you guys hear me?
Yes, and yes.
Right on. Well, hey, this has been informative. Thanks for hosting and pulling it together. Great to see a lot of people on this call. I agreed to summarize habitat conditions in both Idaho and Southwestern Wyoming. And I want to acknowledge co-authors from Fish and Wildlife Service in both Idaho and Wyoming who helped pull together photos and a little bit of information on habitat composition. I'll note that our quote strongholds in Idaho and Wyoming would be considered relatively weak places further south. So we have some sites that have been occupied off and on, and sometimes on most years, sometimes sort of more off and on over many years. And those include all of the stars on this map, including Seed Skiddy National Wildlife Refuge in Southwestern Wyoming, multiple sites along the Snake River in eastern Idaho, and this site along the Big Wood River in south central Idaho. And so I'll be talking about, or at least presenting photos from each of these sites to give a sense of habitat structure. And then we'll also add some information about Fort Boise Wildlife Management Area in southwestern Idaho, right at the border with Oregon, where the Boise and Owyhee rivers flow into the Snake River, where we actually got a detection or a couple detections this summer. So I just wanted to show this map for sort of like landscape context, a lot of desert, a lot of mountains in some areas nearby, but also a lot of agricultural land that is often nearby to the riparian habitat that we do surveys in. And I'm going to start with information on Seed Skiddy, which I actually haven't been to. So thanks to Katie and Colin, as well as Mackenzie, who was our field technician doing surveys this summer for some of these photos. This was an occupied site this summer. There was a single detection as part of the Rangewide survey that John presented about earlier in the morning. But there is also a history of sightings, especially over the last couple of decades. And there's kind of a mix of habitat here, it seems like, with some areas with a cottonwood overstory that can be rather sparse actually, but also variable understory. And so here's one shot, and this is actually quite near where the detection was in 2022. And then here's another picture from sort of the side of the river, giving you that desert context with cottonwoods and some shrub understory. And then just to step back and look at an aerial view, just to give you a sense of how much desert surrounds the Seed Skiddy National Wildlife Refuge and the Green River that flows through it. It really is all desert in this area surrounding it. And then if we zoom in a little bit to get a sense of aerial imagery, you can see there are cottonwood trees along and right next to the river, but oftentimes rather sparse in terms of their coverage relative to other areas where I've seen cuckoos anyway.
I wonder where they're nesting.
And then this is an interesting shot, just sort of from a bluff above the river that, gives a view of some really nice looking habitat, but still rather sparse cottonwoods, but a more developed understory here. And so this is an area in Wyoming that has had the most consistent track record of sightings over the years. I'm going to shift quickly to Idaho and try to capture some of the landscape context. And so I have that in the notes on the side, but I might just walk you through a few photos showing some spots in Idaho. So this first one is from a high point overlooking the best stretch of habitat we know of along the Big Wood River, which does flow into the Snake River. And this is in south central Idaho. For many years, there have been 1 or 2 birds detected in this area as part of formal surveys. And then before the species was listed as threatened and playback was stopped for general birding community. It was a place that birders often would go and try to find them. But we've never found more than 1 or 2 birds here. And so it's a rather disjunct area. It looks amazing in terms of overstory and a mix of understory, but probably not a kind of patch that could support more than a couple of pairs and pretty separated from some other good stuff. And then a couple of shots along the Snake River in eastern Idaho, which happens to be our quote stronghold. And where we've had the most detections and the most survey effort over the last 20 years or so. That includes Idaho Fish and Game, both doing surveys and funding surveys with consultants and our bird observatory. But I will say, just for context, IBO, our bird observatory, has been doing surveys since 2018. So 5 consecutive years of pretty intensive surveys. And in 2018 and 2019, we had relatively decent detections. In the last few years, it's been less and less. And actually 2022 seems like the fewest we've seen in all those years. And so there's a dynamic going on, whether that's, regional variation and, fluctuation that might bounce back with different environmental conditions, or hints of a decline, I'm not sure. But anyway, back to these pictures. So here's a shot looking at the Snake River, relatively far upstream, relatively close to Wyoming, with some nice understory that you can see in the foreground, oftentimes willow, some dogwood, oftentimes some Russian olive, but in many cases, it hasn't been the dominant species, and then some cottonwood and even some conifer trees sort of on the edges. And then here's another shot upstream of American Falls Reservoir. So the furthest sort of southwest of our southeastern Idaho stronghold. You've got a moose and a couple calves crossing here, sort of between a couple of different patches of habitat. And you do see some Russian olive and other understory veg with some cottonwoods here. And if, assuming Katie Powell is on the line, I'm going to hand it over to her for a moment. Sorry here to surprise this on you, but Katie's going to talk for a minute or so about this Fort Boise Wildlife Management Area, which has not been traditionally part of our survey area, but was fortunately selected as part of the Rangewide survey this summer. And even though we had some detections in other historical sites in eastern Idaho this summer, none were actually on the site selected by the statewide survey, except this site, well away from the other site. So Katie, if you're on, feel free to jump in, and I can advance the slides for you.
Okay, great. Can you hear me okay, Jay?
Yep.
Okay. For those of you who don't know me, I'm Katie Powell. I work for the Fish and Wildlife Service in our Ecological Services office in Boise, Idaho, and I'm the yellow-billed cuckoo recovery lead for the service in Idaho. And I was lucky enough to be able to survey for cuckoos at the Fort Boise Wildlife Management Area this summer. Like Jay said, it's located on the border of Idaho and Oregon, where Idaho side Boise River flows into the Snake River, and where the Oahe River flows into the Snake River on the Oregon side. And all of this riparian habitat in the WMA is surrounded by agricultural farmland. So there have been very few detections, kind of anecdotally, over the past, I think, 50 years, and it has only been semi-formally surveyed once about 10 years ago by the BOR, where they did have one acoustic detection in mid-June. We were, hopeful for detections, but not optimistic. Again, not a stronghold of habitat. The patch size of cottonwoods in the area compared to the habitat that we have in eastern Idaho, where we have identified critical habitat, is, much smaller. So we were very excited to have acoustic and visual detections in both the first and second round of surveys here, which could indicate a breeding pair or two. So cottonwood is the main overstory here, with willow and locust as the main understory species. All of our points were located near to water, mostly free flowing along the Boise and the Snake Rivers. If you want to advance, Jay. So our points kind of centered around, even though they were on these, along the Boise and the Snake, along free flowing water, most of our points kind of centered around this very stagnant, swampy, so smelly area where the Boise meets the Snake. And if nests are present, we think that it's probably kind of in the depths of this area. So maybe next year we can get out for some nest surveys. Really exciting though to have detections in 2 rounds in an area where we weren't expecting. And to see birds, not just hear them, but to see them.
All right, thanks, Katie. Yep. So here's a shot of Katie's colleague Jan during their playback surveys, very near where they had detection. So you can see a mix of some lower shrubs, some mid-story, but also some cottonwood in the background. So seemingly really good habitat, but again, a patch issue where how much good habitat is nearby that could support actually a population of cuckoos. And just before I close, I'll talk a little bit about patch characteristics sort of while we're on that subject. And so here's a section of our surveys that we've done. This was in 2021, so this wasn't this year, but showing the designated critical habitat. So this long stretch along the Snake River from the southwest part of the picture or the image which is at American Falls Reservoir upstream for about 20 or so miles. And so we surveyed a lot of that. And so there is this rather contiguous patch there. Then there's kind of a break upstream as you get further up towards the northeast part of the image, and even further up where there's a very narrow riparian stretch. And then upstream further, you get into a few other places that are semi-contiguous, but there's also some relatively isolated patches of really well-developed understory and midstory. So just a couple of thoughts about patch dynamics. And I'll just mention, because we have a relatively small sample size in Idaho and Wyoming and relatively few detections, we've actually put some effort into nest searching, unfortunately coinciding with our worst years of detections. And so that didn't pan out for us. So we can't say a lot about nesting and nesting habitat, just more the habitat where the birds are found. So I might just, in the interest of time, pause there. Maybe there's a minute for questions, but we'll have some time to discuss this later. Thanks.
Jay, you do have one question in the chat, which is, are you using the model to direct your surveys?
Yes, and so both, I guess I could just speak. So this year, as part of the Rangewide survey, we used the model that John talked about, and we selected sites there. So we had 12 sites in Idaho, including what Katie just described. In previous years, Matt Johnson and Jim Hatton had helped produce a model for Idaho, and we've used that to select our survey sites going back to 2018.
Excellent. Thank you for that.